So, as my clock ticks over to 11:33, I suggest we start. A little while ago, I was absolutely thrilled that we had 30-something people who joined the call, and I can see that we're now up to 46. And now I'm feeling intimidated, Alex, because you told me that 180 might join us. However, we have got quite a long meeting planned today, and I predict people might dip in and dip out of today. So first of all, thank you very much for joining us. My name is Jeff Hall. I guess I'm here in two roles. The first of which is I'm the Chief Clinical Data Officer at HDRA UK, so have been chairing the special interest group that has been meeting to discuss OMOP for some time now. But I guess that my second declaration of interest is that I'm a medical oncologist by trade. So my personal interest in OMOP has always been within cancer. And at one level, I'm quite impressed with myself that I've managed to leave it until our 7th special interest group, until we've had a meeting focused on those two worlds coming together. So I'm delighted today that we are talking about cancer and the OMOP Common Data Model. And what's great is that we're going to be able to hear from a range of people at a number of centres across the UK. And we've also got some an international speaker joining us to talk about the worlds coming together. Now, my fellow chair, Danny Prieto-Alhambra from Oxford, is unfortunately not able to join this first part of the call because I think he's at an international OHDSI meeting. Is that right, Alex?
I think it's a Darwin EU meeting.
Okay, so we'll have to accept that's a reasonably good excuse to not be here at the moment, but he is joining us later. So I hope that most people who've joined today have got a working idea of what OMOP is. And therefore, I don't suggest that we spend a lot of time doing the background to OMOP. But at the end of the day, here we are to discuss the concept of a common data model that can work locally, regionally, nationally, and internationally to facilitate large federated studies of clinical data, and in particular, clinical data relating to cancer. So as we now have a reasonably good audience of more than 50, I suggest we dive straight into the first item on the agenda. And I'm delighted that Charlotte from KCL and Andrew from Oxford are going to talk to us about the Cancer Research UK data community. Charlotte, can I give you the floor? Alex, are we recording this call?
Yes, we are.
So we are recording this call, so people who aren't able to join us now can join us later. We'd also appreciate if people could keep themselves on mute unless they're specifically asking a question, and we'll try and find time at the end of each talk for questions. But I'm also happy that questions are added to the chat, and we'll do our best to keep an eye on the chat and make sure those questions get asked through. But Charlotte, do you have slides that you wish to share the screen?
Yep, I can share my screen. I'll just do that
over
now.
to you.
Thank you. Hopefully everyone can see my screen and it's in PowerPoint presentation mode. Amazing.
Yep, perfect.
Thank you. So as Jeff mentioned, I'm Charlotte. I am a postdoctoral research fellow from King's College London. I work in the team of Professor Mika van Helmerick, who is a cancer epidemiologist. And I'm going to be talking a little bit today about our CRUK Data Community Support Unit project, which I'm working on with my colleague Jasmine Hanford. And we're also working closely with Andrew Blake from the University of Oxford. And he was actually billed to speak with me today, but unfortunately something has come up. So you just have me today. But we are working on a project that is looking to develop a strategy to standardize and allow the safe and efficient reuse reuse of CRUK data sets, so existing data sets that have been collected in a previous context across the Cancer Research UK setting. So I just wanted to start by giving a little bit of background as to the Cancer Research UK research data community. And this is a community that has been set up with 6 pillars, and it essentially aims to support a connected and collaborative research data community that will allow us to identify data-led research questions, access NHS and real-world data, build multidisciplinary teams, and develop training and early career support, as well as undertaking exemplary PPIE and EDI around data. Essentially, this works to allow us to future-proof data sharing infrastructure, instill data fundamentals, and then essentially develop novel and innovative data methods. And as I mentioned, the CRUK Data Community Support Unit is formed of 6 pillars, which are led by different individuals across the research community. So we have the data and compute infrastructure, public involvement and diversity, training and careers, data and samples reuse, spatial biology, and then health systems data. And we're always looking for people who are interested in joining any of the special interest groups. So if you are interested, please find a sign up link on the CRUK website. But a little bit more about the background of this project. So we know that data has become a key asset for the economy and for our society, and the need to make sense of big data is leading to innovations in technology. And advances in data-driven precision medicine are underpinned by understanding disease subpopulations using rich medical data, as well as advanced analysis methods. And we know that cancer data comes from diverse sources, so from electronic health records, pathology reports, biomarkers and omics, imaging, and then emerging patient-reported outcomes, so things like health-related quality of life and devices. But key issues that exist at the moment include data heterogeneity. So we know that data varies greatly in structure and format across sites, which then makes it difficult to bring the data sets together and essentially kind of impedes comprehensive analysis. And large data sets are needed for robust cohort analysis, but these are often also unavailable. So a solution is to pull data sets from multiple trials and studies together, standardize these data sets using a common data model such as OMOP. But essentially another barrier is that legal and practical restrictions, so things such as contracting and the need for data sharing agreements, prevent this pooling in what we call a centralized model. So this impact is it limits the integrated use of data, slows our progress in data-driven medicine, and this is especially true for rare cancers where data availability is often more scarce. So the idea of this project is to kind of facilitate the reuse of existing CRUK data sets that have been collected previously for alternate purposes, and to take these data sets and standardize them using the OMOP Common Data Model, but also then enable collaborative research using what we call a federated data model. So a federated data model differs from centralized models. So in a centralized model, each site would essentially contribute their data to a central site, and that central site would then do all of the analysis. But that requires kind of data sharing agreements, rigorous data governance procedures, and it's very time consuming and resource heavy. So a federated data model kind of overcomes this by allowing each site to kind of maintain their data at their site. And then what happens is analytical tools are deployed by each site, and then aggregated data, the aggregated results are shared. So our project aims to focus on expanding OMOP adoption across the Cancer Research UK setting, and we aim to provide training and resources to facilitate this. It also aims to develop data sharing strategies that are tailored for cancer data sets, and then drive progress essentially towards kind of reaching the CRUK research strategy goals, so fostering collaboration, enabling large-scale data integration, whilst also ensuring privacy and compliance by employing these federated data models. And as I mentioned, this then kind of aligns with the overarching aim of the cancer research data community, in that we aim to accelerate data-driven cancer research and then ultimately improve patient outcomes. So our project is formed of 3 work packages. So the first work package is one that myself and my colleague Jasmine are working on at King's College London. So we have been working to develop a strategy for adoption of the OMOP CDM and also sort of the federated data models across the CRUK landscape. Concurrently, Andrew Blake's team at Oxford are working to develop the infrastructure that is required to begin to transform some of this data, so standardize the data according to the OMOP CDM, and then also develop some of the sort of analytical tools and the infrastructure that's required to use this federated model of data sharing. And then work package 3 will essentially take the kind of output and learnings from work package 1 and work package 2, and we aim to develop and disseminate a sort of training modules and education across the Cancer Research UK space. So work package 1, we have been undertaking what we call a SWOT analysis, so identifying strengths, weaknesses, opportunities or threats or barriers and facilitators for OMOP adoption and data sharing and access across Cancer Research UK funded projects. And we have developed a kind of mixed methodology. So we've been undertaking various focus group discussions over previous months, but we've also developed and disseminated a web-based survey using the REDCap platform. And we've had input and assistance from various networks and groups as part of kind of the Cancer Research UK data community, but also more further outreaching. So we attended the UK Oncology Forum meeting back in June. Jeff was in attendance, and we kind of ran a sort of live focus group discussion at that meeting with healthcare professionals. We've also recently in October undertaken a focus group discussion with patients and patient representatives from the CRUK Patient Involvement Network. We had 11 patients and patient representatives in attendance. It was really a fantastic meeting. We're also in contact with the OHDSI Oncology Working Group, so we aim to attend one of their research and output meetings, either the December meeting or the January meeting, and we'll kind of run through some of the output to date, but also run a live focus group there. And very helpfully, we've been in contact with Heron UK Network, and they have disseminated our web-based survey as well, which has been really useful. And I just want to give a kind of very brief overview of some of the sort of prompting questions that we've been using in the focus group discussion, but are also included within the survey. So for strengths, we're asking things around like what are the key main benefits of reusing information that's already been collected as part of previous studies, weaknesses, and what would help make patients feel reassured that their data is being standardized in a safe and trustworthy way. In terms of opportunities, do respondents think that this could make research fairer or more inclusive in any way? And in terms of threats, would you have any worries about whether the meaning of the data could get lost or change when it is standardized? And just a bit of a shameless plug, our web-based survey is still open, and so if any of the attendees today would be willing to complete the survey, it would be much appreciated. It can be accessed either through this QR code or via this link, and I will see whether or not I can get this disseminated after the meeting today as well, in case we can get any more respondents. But to date, we have had 36 participants who have contributed today over both the focus group discussions and also the survey. This is formed of 7 researchers, 11 patients, 7 data focus roles, and 7 other supportive roles. And we're kind of lacking in healthcare professional respondents at the moment. So if there are any clinicians or healthcare professionals on the call, please do take part and please do complete the survey. It would be much appreciated. But what I can say is that we do have representation from across the devolved nations, which is amazing. And also it's been useful because those who have taken part have had a varied knowledge and experience of the common data models and federated models of data sharing. So I really feel like we've got a kind of full picture, which is great. So I just want to run through some of the sort of strengths, weaknesses, opportunities and threats that have emerged. So we have been undertaking more qualitative thematic analysis of the focus group discussions, but also the survey responses as well. So some emerging strengths are around the huge potential in maximising the use of data, particularly in the NHS setting, that there is good governance available to facilitate OMOP CDM. This will allow us to ask the same question across different data sets easily, and it also facilitates collaboration across various health organisations without the need for rigorous data sharing agreements. In terms of weaknesses, a kind of key emerging weakness is that there's potential for certain sites to be left behind or negatively impacted if they don't have the resources or finances or sort of technical infrastructure in place to facilitate this. There also is a lack of tailored and dynamic training to support data standardization and sharing at present. As I mentioned, the lack of resource and or funding for data teams to manage the OMOPification of the data. And then also the fact that the OMOP CDM vocabularies are dynamic and kind of ever evolving, so that we need to ensure that there's sort of sustainability funding in place to enable that long-term kind of data mapping and updating of the data sets according to changes in the OMOP vocabularies. In terms of opportunities, it's been said that it would save resources, but also patient interaction in terms of sampling. So if patients have been recruited onto a previous study and then we can reuse that study data, it then saves resource and then sort of patient time and input. It will facilitate research on a more worldwide level, translating into better outcomes for patients, which is obviously one of the key aims of the Cancer Research UK research data community. It will allow us to see gaps in available data and then focus studies accordingly. So develop studies that then work to fill in those gaps. And it should enable us to develop AI models from a large pool of clean, structured and standardized data. In terms of threats, this was a big one that came out in the patient focus group. So the need for kind of transparency and the potential need for multi-level patient consent. So if patients have been recruited and consented into previous CRUK studies and their data's now being reused for alternate purposes, we might need to look into kind of reconsent. Access to new systems and processes will of course be required. And then historic or current lack of recording of certain measurements will lead to holes in the data. And someone mentioned that the complexity of mapping data to the OMOP, that user stories would be useful. So this has kind of led us to where we're up to now, which is beginning to sort of develop the strategy as part of Work Package 1. So some of the emerging things for this strategy include promoting collaboration and governance frameworks, so building on existing governance and CRUK networks to reassure stakeholders about safe and trustworthy data standardization and sharing. And then, as I mentioned, one of the threats that emerged was the usefulness of kind of use cases, user stories. So we aim to highlight the benefits of the OMOP CDM across the Cancer Research UK setting by showcasing real-world examples of how OMOP enables cross-data set analysis and then accelerates research. We also aim to develop tailored training programs, and these will be modular role-specific training that will address knowledge gaps in OMOP.
Forgive me. Oh,
sorry.
Let me just mute somebody. There we go. Charlotte, sorry, carry on.
That's all right. So yeah, this will be modular role-specific training that will aim to address knowledge gaps in OMOP and federated data sharing and models across different roles. As I mentioned, it will be really important for us to secure sustainability funding, so advocate for long-term resource allocation for OMOP mapping and vocabulary updating throughout CRUK. And then clarify the handling of real-world data types, so establish international guidelines for standardizing nuanced variables to ensure data integrity moving forward. We also aim to try and facilitate global collaboration, so pilot this federated learning to test international research without the need for its setup of extensive governance, and then undertake a gap analysis for targeted study development. So creating kind of catalogues or dashboards that demonstrate data availability to then inform and prioritize future research. And essentially, we want to position the adoption of OMOP as a foundation for facilitating the use of AI and advanced analytics. And finally, as I mentioned, it will be really important for us to kind of assess the consent models that are currently in place for these CRUK data sets, and then develop clear multi-level consent frameworks to address data reuse and any confidentiality concerns that these patients may have, and then address potential legacy data issues. So develop strategies for handling incomplete historical data, so things like multiple imputation or annotation of the data. So in terms of next steps of this project, so Andrew and his team will be working to pilot OMOP conversion. So they are in the process of selecting some CRUK pilot data sets which incorporate various data types. They will then map the data to the OMOP structure and then convert these original data sets into the OMOP format using the ETL process. They'll perform quality checks and validate to ensure integrity and context, but then an important thing for them to do will be to document the challenges, what resource was needed, and the time taken to pilot this standardization, so that we can kind of put this together with our findings and really nail down a strategy that is going to be far-reaching and applicable across sites. So what I mentioned is our next steps will be kind of work package 3. So that will be a combining work package 1 and work package 2 findings to develop a training hub that will include webinars, interactive tools, and case studies, developing a tailored governance toolkit for federated data sharing, addressing that multi-level consent frameworks, and then analytical toolkits that are required to facilitate federated data models. And also the kind of key thing is engaging stakeholders from across CRUK and the NHS, right from early involvement, but throughout this project to ensure inclusivity. So if you are interested in taking part in this study or finding out more about the output, then please feel free to reach out to either myself or my colleague Jasmine. Our email addresses are on the screen, but we will look to disseminate some of the findings across this network if possible, because I think it's quite applicable. Although we're doing it in a CRUK setting, I think this will be applicable across NHS settings and research settings. Thank you very much for your time.
Charlotte, thank you so much for that. I can't underestimate the importance of CRUK engaging with this process. They're an enormously trusted brand within cancer research. And I think cancer research's active participation and leadership in this space is absolutely critical. And, you know, we're sitting in a group that has come together under HDR UK, Health Data Research UK, and this for me is the perfect coming together of HDR UK and CRUK working collaboratively to deliver cancer in OMOP studies. So thank you for that. One of the first questions that came up as you were speaking, Charlotte, was can we put the survey link in the chat? And Uwe has spotted that and has put the link to your survey in the chat.
Thanks so much.
I'd really encourage an audience of 78 people to click through that link and respond to Charlotte's survey link, because I bet you that the collective experience and wisdom of people on this call could greatly add to that survey. So there is a link in the chat. I'd encourage people to click it at some point today, whenever, and help. Are there other people who have other questions that they want to put? Alexis.
Thanks very much, Jeff, and thanks for that fantastic presentation, Charlotte. I was just curious if, given the development of the strategy, I realize you're kind of in the information gathering phase and you'll start to develop the strategy, but I'm curious if there is an expectation or a desire that kind of future CRUK funded projects would be required to use a CDM like OMOP in how their data is maintained and preserved, because I know CRUK is very interested in data reuse, and they obviously have the Cancer Research Technologies arm that are kind of putting data out there for reuse. So yeah, just curious if you think that would be an outcome of the strategy development.
I think that is potentially, yeah, I think that will be a output. And we've been in discussions with some of the other members of the community support unit. And that has been one of the kind of key points of discussion in terms of setting out that expectation as projects are funded through CR UK, that they will be expected to standardise their data into the OMOP CDM format to allow its reuse down the line.
I think that's a great idea, and I'm really happy to hear that.
Yeah, Alexis, I would echo that. I think it's through leadership of research organisations that have, in many ways, mandated the use of the engagement of patient and public involvement in research, that none of us would now consider doing research projects without that. And I think if CR UK was to begin the process of, on your funding applications, what are your plans to generate an OMOP version of your data set and make this data set available for reuse, and making it quite clear that if you say, not using OMOP, not planning to allow data reuse, that's very negatively marked in the funding application. I think is something that it would be great to see CR UK's leadership. Excellent. Are there any other questions that are going to come from the floor? If not, Charlotte, thank you very much for that talk. It was great, and I'd encourage people to click through the link and also to pick up those email addresses at the end, because I sense this is a community that the more people engage through organisations leading this, such as the CR UK group, the better.
Thank you.
So, Charlotte, thank you for that. And in the absence of any other questions or comments, I'll take us on to our next talk. Is Cheryl with us? I've not seen Cheryl's name. Bingo. Hi, yes. Cheryl. Thank you for joining us. So Cheryl is going to dive into the nitty-gritty of one of the studies that's underway at the moment. So Cheryl, I understand that you're going to talk to us about the IHI Optima study, which is target trial emulation of surgery versus radiotherapy in prostate cancer. And thank you so much for offering to give that presentation. And I'll hand over to you. And I assume you have control of the talk and can present slides, I hope.
Yes, I'm just sharing my screen now. Could you see that?
Yep, but we're not in presentation mode yet.
Okay.
There we go. That's perfect.
Thank you for the opportunity to speak at the symposium today. My name is Cheryl. I'm a senior researcher in pharmacoepidemiology at the University of Oxford, and I'll be presenting one of the IHI Optima projects today. So before we start, I'd like to give some background information about Optima, and as well as the umbrella of the Innovative Medicines Initiative. So IMI is one of the world's largest healthcare public-private partnership, and between 2008 to 2024, it had invested 5.3 billion euros in pre-competitive research. So this is funding that's from the European Union, together with FPIA, which is the European Federation of Pharmaceutical Industries and Associations. And from it, there's born a lot of different projects with project outputs and publications. So in this ecosystem to promote the translation of science and research. And this ecosystem supports the cooperation between public and private sectors, so between patient groups, academia, pharmaceutical industry, regulatory bodies, startups, and clinical experts. And since 2022, this IMI has transitioned to being called Innovative Health Initiative. So there's a more broad focus on other healthcare aspects rather than the initial focus, which was more on drug development. And Optima, the kind of acronym, it stands for the optimal treatment for patients with solid tumors in Europe through artificial intelligence. The vision is to enable oncology patients to have access to kind of most up-to-date individualized treatments and innovative therapies. and how this collaboration aims to achieve this is using data-driven approaches by integrating real-world data and using AI models to improve patient outcomes. So this project started, the funding period started in October 2021. It's for a duration of 5 years, so we're in the last year of our funding now, and it has 38 partners across 14 countries in Europe. And the project focus is on lung, breast, and prostate cancer. So there are 9 work packages within this project, and University of Oxford is co-leading work package 6, which is on kind of data gathering and access. And we also use the OMOP Common Data Model. So there are data partners that come into the project and mapped their data sets to the OMOP CDM. So there is work package 1 that is project management, work package 8, which is involved in disseminating and communication of any outputs from the project. Work package 2, which is the ethics, governance, and regulation. So they are in charge of all the legal documentation in like bringing all these different public and private partnerships together. And then we have work package 3, which are the group that will look at the knowledge gaps and what research questions to prioritize, and also developing a guideline-based decision tool. And then we have work package 4 and work package 5, which are involved in the technical and infrastructural requirements and implementation of the platform. And work package 7, which is leading on the AI knowledge-based implementation. And lastly, work package 9, which is looking more at sustainability of the project. So as mentioned before, the research areas of interest for Optima is prostate cancer, breast cancer, and lung cancer. And this is done through development of research questions surveys with stakeholders involved, so including also healthcare professionals and patient representatives, assessing the clinical guidelines for these cancers of interest, and then to identify the guidelines and pathways most commonly used by clinicians. So with the prioritization of these critical research questions and knowledge gaps, then we go on to this would feed the research questions that we move forward with. So the data that feeds into the platform, there is both, we allow both a federated approach, and also there are central data sources, although most of the data partners here have the federated approach due to the more complex data governance issues. So all the data sets would have been mapped to the OMOP CDM. And then if the data source has a governance that allows for it to be uploaded into the central platform, then it's there for certain authorized users to use them. For the federated data sources, it's will disseminate analytical packages, and then aggregated results are returned to the platform and uploaded there. And within the platform, there are also the other work packages that's working on the more like to input computer interpretable guidelines, and also that's the platform we could have host the any analytical tools or AI models there. And in the end, the output is to facilitate knowledge generation to fill in the knowledge gaps that has been identified. So as part of the data gathering process as well, we had run this diagnostics phenotyping package across all the 18 data sources. So we could see that there were 46 million patients in observation, and from the broad cancer phenotypes that we have for prostate, lung, and breast cancer, there were over 2 million patients in these broad definitions. And it is a mix of some cancer registry data, population-based like GP data, some biobank data, and also some pharmaceutical clinical trial data sets. So you could see for different data sets, there is kind of a different proportion and distribution of cancer patients, because some data sets, they are just for a particular cancer of interest, whereas there are other data sets which are the more like broad GP databases, then they would have a distribution of different cancer patients. So now I'll jump into the use case, the title of the talk, which is to look at the prostate cancer radiotherapy versus prostatectomy. So some background of this study is born from this PROTECT trial, which stands for Prostate Testing for Cancer and Treatment. This was a pragmatic clinical trial that was conducted in the UK, and there were two components. The first component was to test on screening. So between 1999 to 2009, there were men who were invited through their general practices to undergo screening. So between the age of 50 to 69, they received a PSA test. There were about 82,000 men that participated in this phase of the study. And from this, about 2,600 men had localized prostate cancer diagnosed. And then going forward, these men were offered in this second component of the clinical trial, 3 different treatment arms. So they were enrolled in this trial. There were about 1,600 of them, and it was randomly assigned to 3 treatment arms. So there was the active monitoring arm, prostatectomy, and radiotherapy, and there was about 500 or so patients in each of these arms. So because there are restrictive eligibility criteria in clinical trials, they could underrepresent patients in clinical practice who are older and less healthy. Therefore, it is important to understand the difference in the patient characteristics between those who receive treatment in clinical practice versus the trial population, and this could affect the generalizability of the trial results. So the objective for the study is to describe and compare the characteristics of patients who received radical prostatectomy or radiotherapy in real-world data versus target trial in the UK. That's the scope of the presentation today. Well, of course, this study has the wider aim of doing the comparative effectiveness and safety analysis as well. So for the data source, we use the clinical practice research data link, so CPRD, ORAM, and GOLD primary care data. And this would have been linked to the hospital admissions, the HAS admitted patient care data, and also cancer registry, the tumor registration data set, and also the radiotherapy and chemotherapy data sets. So we used the target trial emulation approach that was introduced by Hernan and Robbins. So we used the criteria based on the PROTECT clinical trial study protocol to define our target trial and real-world data populations. So this going through this target trial emulation approach will help us to minimize any biases during the study design phase, and also we could compare the differences in our eligibility criteria, what we can emulate when using routine clinical practice data, and this would help to contextualize our findings. So this is a study design for the broader real-world data population. So the index dates would be the first radical prostatectomy or radiotherapy treatment that was recorded in the data. And then we included male patients who are aged 18 years and above with at least 1 year of observation. And we excluded patients who received, who had either prostatectomy or radiotherapy recorded on the same day. We included patients with early stage prostate cancer, so with the TNM staging, the T1 or T2, which is the tumor size, and not having any metastasis. For the washout window, they should not have any prior prostatectomy or radiotherapy. And then we assessed the medical conditions and measurements anytime before index date, and also medications in the 1 year before index dates. And we will follow up the patients until end of registration or the end of the study period or death. So for the target trial population, it is quite similar criteria, with the exception that the prostate cancer diagnosis was we limited between the age of 50 to 69, and then they also had to have a PSA in the range of between 3 to 20. That's following the PROTECT clinical trial definition. And then there are a lot of additional exclusions, as well, as you would expect from a clinical trial. They will try to include a healthier population. So they will exclude other types of cancer, having kidney dialysis or transplants, or things like bilateral hip replacement, and also having diagnosis of chronic obstructive pulmonary disease, heart failure, stroke, or myocardial infarction in the year before receiving the treatment. So these are some of the preliminary results that we have. So in the columns, you could see the 4 different groups. So there is the prostatectomy target trial group, the radiotherapy target trial group, and then the prostatectomy real-world data group and the radiotherapy real-world data group. And you could see from the table here that the target trial groups, because of the more restrictive eligibility criteria, then there is a lower number of patients in these groups. And those who received our prostatectomy and radiotherapy in the real-world data were older than the target trial population, as expected, because there was the age limits in the target trial population. And for the radiotherapy group in particular, we also observed a larger difference in the age. So you could see it's 72, median age of 72 versus 65 in the radiotherapy group, as compared to 65 versus 62. And also there is a higher percentage of them with the Gleason score. So Gleason score is a grade. So the higher the score, the higher grade of cancer that you have, and then that is a worse prognosis for the patients typically. And for the radiotherapy group, you could see that there is 22% in the real-world data population versus the target trial population, which is 14%. And the percentage in the prostatectomy group was more similar, with 11 and 10%. And then we also see in both the target trial and real-world data cohorts, patients who received surgery or prostatectomy had fewer comorbidities. And patients in the real-world data cohorts received more drugs for benign prostate hyperplasia and obstructive airway disorders than the target trial cohorts. And again, for the radiotherapy groups, there is a larger difference in the percentage with the comorbidities and medications versus the prostatectomy group in real-world data as compared to the difference in target trial. So for example, if we look at the hypertension row, there is 57% of patients in the radiotherapy real-world data group versus 46% in the target trial group. This is a higher percentage in this comorbidity as compared to the prostatectomy group, which is 36% and 40% respectively. And when we compare this with the PROTECT trial participants, we see that the patients for the radiotherapy group in particular has an older median age. And then also in our study at baseline, most patients had higher grade cancer in our study. In the trial, they had lower grade cancer. There was 76% in the prostatectomy group and 78% in the radiotherapy group in the clinical trial. And also the median PSA was lower in the clinical trial as compared to our study population. And I'll just highlight that this baseline characteristics is before we did any matching, but we have proceeded to matching, although the data is not shown here, and it's also very similar results that we see even in our target trial population. It is slightly older and having worse prognosis than the actual trial population. So in summary, we see that the patients receiving prostatectomy were younger and had fewer comorbidities than the radiotherapy group in both the real-world data and target trial cohorts. And those who received radiotherapy in real-world data had more advanced disease than in the target trial group. And particularly for the radiotherapy group, we did observe larger differences in age, comorbidities, and medications versus the prostatectomy group than the difference that we see in the target trial population. So the next steps for our study are also outlined in the protocol. So you could scan the QR code to see the protocol that we've registered in the EMA real-world data catalog. So we are going through an iterative process of kind of looking at to emulate randomization. We are doing propensity score matching and also looking at before and after matching how the balance of patient characteristics are like. And this is ongoing work, so watch this space for any updates. And come to the end of my presentation. Thank you so much for your attention and happy to take any questions.
Cheryl, thank you so much for that. That's really interesting to see the work that you're doing. And I'm really impressed. Are there questions from the floor for Cheryl? Cheryl, are there opportunities for cancer centres around the country to contribute their data directly to your project?
Yes, definitely. We're interested. I think now Optima has set up kind of like third-party agreements to beneficiaries that are not within Optima. So I'm definitely happy to have a chat with anyone that's interested, because the data set that we have currently is mainly English practices. So I think if there are kind of and also I think CPRD, the cancer registry, is limited to a certain time period as well. So we are happy if anyone would like to participate, especially with more recent data sets.
And Cheryl, would you hope that centres can contribute data or can run an analysis against their data set?
I think running analysis, yeah, because I understand all the data governance with, contributing data is quite difficult, but we have already, at least now we have the diagnostics that's ready to run. And I think once we have confirmed the study analysis code, the study analysis package would be ready to run as well.
Fabulous. Are there other questions from the floor? So while people are thinking whether they have other questions for Cheryl, there has been a chat going on whilst Cheryl's been speaking, and I can't but help but come back to your comments, Nicholas Richmond from NHS England. Nicholas, are you still with us?
Yes, hello, hi, I'm here.
Brilliant, because the talks so far have raised the questions about cancer registries and one level, their use of common data models. And it remains an obvious question. I must stress that perhaps my first interest in this was the day I realized that English and Scottish cancer data models are different and therefore can't be easily aligned. And then as I've run international cancer data studies with other countries, the crying out need. So I wonder whether you just wanted to say a little bit about the program lead for the development of an NHS canonical data model and its relationship to OMOP.
Sure. So this piece of work has been underway for a year or so. It is predominantly to bring together a collection of national data sets that we've been gathering for a number of years, which have it's always aligned to the NHS data model and dictionary, but they still have some differences between them. But we're taking this opportunity as the development of the federated data platform, which you might be sort of familiar with, which is currently being rolled out, and we're bringing all of our data assets onto a single platform, and the benefits of creating a single data structure to define all of the information that is held there. Predominantly, it is patient level information that comes directly from the electronic patient record systems submitted on a monthly basis, but we're looking to move to more event-based data flows. But the data structure it's sort of built on a lot of previous work. So we're developing a single logical model that describes the subject of care or patients, but it's also will cover social care. So it's slightly broader than that. And it includes all the elements you'd expect around the patient information, the service request, contacts and activities, diagnosis, inpatient stays. It follows a patient administration pathway and then is supplemented by clinical terminology, typically using SNOMED CT and other clinical terms. It also has a workforce lens, focusing on the staff and information around there generated from the electronic staff record, a lens that relates to equipment and devices, the supply chain, and also a financial lens, which kind of brings it all together to look at the costs. But we are aligning it to a number of existing standards. I've already mentioned the NHS Data Model Dictionary, which largely describes most of the large national data sets, including the cancer data sets that are flowing at the moment. We're aligning it with FHIR messaging for the data in transit, which will be used for populating that information in the future, especially through event-based mechanisms, but also OpenAir for describing how the information is held in the local EPR systems. But we also want to produce a range of analytical assets, physical assets that are based on this large data structure. One of them will be feeding the single patient record, which, if you see in Wes Treating's 10-year review, gets mentioned quite a lot. But the other aspect is the research community. So obviously we're very aware that OMOP is becoming a standard for describing data within the research community. So based on this very low level detailed information, we can produce analytical assets, and the intention is to produce them aligned to the OMOP standard, so that then we can expose that to external companies and research bodies to use that information. So rather than creating something new, we want to make best use of what's available out there already. So that's where we are. We've got the governance in place now. We've developed a number of domains. So by domain, I mean things like a subject of care, or how we describe diagnosis or referrals, that sort of thing. And so those logical models are starting to be published, and you can see those on the link that I provided. But then there is the physical representations of those. We're now going through the national data sets. We're doing emergency care and theatres, and local costing collection data sets at the moment, aligning them to the NHS canonical data model, and then reissuing those standards that would then require the providers to start submitting the data in this structure that aligns to this single standard canonical data model.
Brilliant. Well, I desperately
nutshell.
hope that you've mentioned in the chat again that you hope to align that canonical data model with OMOP, and I hope you do that very closely, because of course all of us learning to speak one language here is much more helpful than us learning to speak NHS England speak for our data, plus then an OMOP language for national and international collaboration with research. So the more they are aligned, the better. And at one level, Cheryl, I can only imagine a future in which all of the cancer demographic data is linked to the cancer diagnosis data and the radiotherapy data set and the HES procedure, well, the OPCS procedure data, such that Cheryl could be running her real world data study against the national data sets for cancer. Am I right, Cheryl, that for English data, you're using primary care data here?
Yes, so how we define the population, I think it was how the data sets were in CPRD at that time. We had to define, because I think the cancer registry data was external to CPRD then. So we had to define our patient population using the primary care data first and then link it to the cancer registration data. But I understand now the workflow has changed a bit, so you can start with a cancer registry population and then work backwards to get the primary care data linkage. But I think it's still limited to up until 2018 for the data set that CPRD has access to, as opposed to, I suppose, with through NHS England, there will be more recent data sets.
So up to diagnoses in 2018, is that, Cheryl?
Yes.
Yeah, I mean, clearly,
data, yeah.
clearly the research community needs this data to be all but real time. So one of the things that did come out of the COVID pandemic was the creation of the rapid diagnosis data set within NHS England. And that is literally up to date within a period of a few months and weeks. So that is the point that I hope that NHS cancer data can reach, because that can really drive and support research. Excellent, thank you. Cheryl, before I move on, let me ask one last time if there are other questions from the floor. And if not, Cheryl, thank you very much for that. That was fantastic. And I'm really grateful that you came and described that work to us. But if I could move us on, is Laura with us?
Yes, I'm here.
Fabulous. So one of the things that has frustrated me for some time is the fact that the large national data sets, cancer data sets, so COSD, SACT, RTDS, etc., have not been available in OMOP form. And then I have become aware of the work that Genome England did, and Laura is very kindly going to talk to us about the work that Genome England have done with cancer data mapping to the OMOP standard. Laura, the floor is yours.
Thank you. I'll just see if I can present my slides. Here we go.
Perfect, that's ready to go.
Oh, excellent. Okay. Well, first of all, hello, and thank you very much for asking me to present on mapping our cancer data. Now, a couple of years ago, we mapped our data from the 100,000 Genomes project to the OMOP Common Data Model. We'd had about 17,000 cancer participants, and we collected some data, which we call primary data from them, but we also had secondary data about them, including NCRAS, National Cancer Registration Analysis Service data. We also have SACT, we have RTDS, and NHS Cancer Registry. As I've only got a fairly short slot, what I'd like to do is talk about some of the challenges we faced when mapping NCRAS to OMOP, as I've had feedback from other people in the field and from talking to people that get-togethers. I found that some of the things that we came across have other people have encountered as well, largely around data that isn't actually in clinical coded format. So it's likely that the things we found will apply to everybody else as well. And if I start getting hoarse and creaky, I've got a bit of a stinking cold, so bear with me. Okay, so just a bit about me. I'm Laura, and I've been at Genomics England for 10 years now, and I've been working with clinical data since 1993. I started off doing data migration to the EMIS system, which makes me feel extremely old, so working on that. I work on the research side of things at Genomics England, and a lot of what I do is involve data curation and analysis. I led on our OMOP mapping work, and we published a set of mappings a couple of years ago. There's a link at the end of the deck. So when I've had enough of that, I channel in a gay advert and play the bass guitar in a punk rock band. Anyway, let's get down to it. Now, in an ideal world, ingesting data from an EHR to OMOP should be straightforward. We would, excuse me a moment. Sorry about that. We would have an ETL tool that would extract data from an EHR system, transform it to the OMOP format, and loads it into the Common Data Model. Depending on what we want to design, we might do some data cleaning and transformation, and possibly even remapping. And we might even be smart and have real-time or near real-time updates, triggers on database tables, loading into a staging table for the ETL to pick up, or something like that. That's the ideal world, and so a lot of the tooling that I've looked at seems to be predicated on that. Unfortunately, in the NHS world, it looks a bit more like this. We don't get the data directly from hospitals. We get it via NCRAS, who do a lot of processing and curation before we receive it. And the same goes for the other data sets. For instance, SACT, if you've looked at the data, omits a lot of the detailed drug regimen information, which makes OMOP mapping quite hard. If you look at the SACT data, you'll see there's a couple of fields for the regimens, but that's just got the drug names bundled up. We don't actually have detailed dose or package preparation, which would make it easier to map to the map to our RxNorm or DMD. We've got enough to do a basic drug era, but it could be better. And a further challenge we had, and this is something that has come up a lot when I've been talking to people, is that some key data points are expressed as permitted values or enumerations rather than clinical codes. But in spite of that, we did manage to map the majority of the NCRAS data. We got an 85% success rate, and some of the attributes that we didn't map, such as coding information, will still be used by our pipelines. Next slide. So we did adopt some guiding principles before we started. The first was to map as much as we could. Now, although our main use is genomic research, and we felt we should provide as much information as possible, as we couldn't second guess what insights researchers might be looking for. Clinical codes were obviously the low-hanging fruit, but the permitted values also had a lot of clinical information. When I looked at some other projects that had mapped, they did taking clinical codes out of NHS data sets. They generally tended to go for the clinical codes, which is, let's say, the low-hanging fruit. But when we looked at actually what was expressed as list permitted values, we could find a lot more. Now, obviously, as we know, in our best practice demand that standard concepts should be used, and we follow this closely as possible. But one big problem we did run up against is that we didn't think it was advisable to add information that could not be safely inferred, which meant that on occasion we had to use non-standard concepts, because the closest standard concept might have qualifiers in the description that we couldn't trace back to the original data set. So here's a very simple example of how we mapped a numeric value. The tumor NCRAS data includes an attribute for recording tumor size. That's in millimeters, but that information had to be drawn from the original data dictionary, as there's no indication of units in the file itself. We've got it's got a file called tumor size, and it's got a value in it. That's it. Right, knowing that, excuse me, knowing that the dictionary told us it was in millimeters, we could easily map it to this SNOMED CT code. So we put it on the bigger screen. Okay, so let's say we've got SNOMED CT code, lesion size. We use type concept of EHR throughout. In practice, there may not, everything might not come straight from the electronic health record, but with no real way of differentiating it. And as type concept is a mandatory OMOP attribute right across the domains, we decided to standardize on EHR with one or two exceptions, but generally we stuck with EHR. Now the permitted values, they need a lot more thought, and here's where things start to get a bit tricky. Now NCRAS can record individual and combined T and an M stages and differentiate whether they're obtained by pathology or diagnostic imaging. And some of the internal information will identify the best values, which are actually recorded separately. Now we wanted to maintain this differentiation. Excuse me. Yeah, sorry. As although we expect pathology findings to be preferred by our researchers, we still want to give them the option. So this is how we did it. So as you can see on the top, the stages obtained by pathology were straightforward enough. We've got a value of 0A that maps straight to a cancer modifier code. But for imaging, we had to amend it to say how it was obtained, both for imaging and we had to do this for best. What we've done here is adding a prefix to the source value by saying T imaging. We recognise it's not ideal to modify source values in this way, but the only other way to identify values obtained by imaging or best would have been to record a diagnostic imaging event and link that via fact relationships. That would have meant inferring information and complicating the linking. We did use fact relationships to link the findings back to the diagnosis code, which is the main event in the tumour file. And I'll say a bit more about clinical codes in a moment. So this works. We get the information to OMOP. It's a bit of a messy workaround, but the information is there without deviating too far from the OMOP standard. And crucially, this won't affect interoperability, because that's something else we have to keep in mind. And it's also caused a few issues when trying to map our genomic data, which we haven't done. So this brings on to how we handle multiple tumours for the same patient by linking on something that can't be mapped. Now, NCRAS has an attribute called tumour pseudo-ID, which is unique to each tumour in the dataset. So we use this in place of some of the source values. So here, we've got two pseudo-IDs, ABC123 and XYZ456, values I've just made up. In reality, they're a lot longer, but this does illustrate the principle. Here we've got two tumours for the same patient. They have the same coded diagnosis and underwent the same procedure, but using the pseudo-ID as a link allows the two to be linked. We couldn't obviously link on dates because diagnosis date and treatment date could very well be different, but this does allow things to be grouped together. So the final thing I should highlight is how we had to combine multiple data points to get a match to the concept table. Now this, unfortunately, is undoing some of NCRAS's work. Now, if you look at the top list, the first three have to be combined and altered. Now, for some reason, the NHS amends ICD codes to 4 characters, either by adding a trailing X to 3 character codes, or in this case, by removing the decimal points between the 4th and 5th characters. Excuse me. These have to be combined and reformatted to find the matching OMOP concept. The format we want is morph coded, followed by a slash, followed by a dash, followed by a reformatted site ICD 1002 field. Now that matches the standard OMOP concept. And the lower the table shows what we had to do. Replace the decimal point to get C04.9, add a slash to the morph coded, add a dash to the behavior, put it together in the right order, and that gives us a map to OMOP standard concept 44502380, which is an ICD-03 code, and that's a standard concept. I mean, we can validate that by checking that both the site coded desk and the histology coded desk appear in the return concept names. That gives us a good second check just by doing checking for regular expression. Those are the main things I wanted to highlight today. I was happy to take questions. You can find our public OMOP mappings in this GitLab repository. As well as NCRAS, we've got HAIRS, we've got radiotherapy, SACT, we've got lung cancer as well. That's a bit old now. We've got mental health, intensive care, the IGNARC stuff, mortality and diagnostic imaging. So I'm always happy to answer questions you may have. We don't have the mappings in machine readable format yet because we're still looking at how best to put a pipeline together. And if you look through some of the mappings, they don't easily map to sort of this source goes to this target. You have to do things like the code reformatting and combining that I've talked about. Some of the HES data sets, HES A&E in particular, needs 3 data points to be checked for body site laterality and that type of injury. But I say a lot of it is we think it's a fairly good rich data set, and if you want to make use of it, go grab it. We're always happy to share, and that's my email address. So please don't hesitate to get in touch if anything crops up, if you have any questions. Excuse me.
Laura, thank you so much, and thank you for battling through your upper respiratory tract infection. No, not at all. I really admire you for just head down and going for it. So I think this work is incredibly important. I think that you have broken really important ground for the rest of the NHS. And at one level, you talk at the end about your GitHub repository and how you can access the mappings. What many people will want to do is access the data, the OMOP transformed version. Is that data accessible anywhere? I appreciate you can't hand out NCRAS, HES and SACT data because it was given to you for specific reasons. But has that OMOP map data been given back to NHS England such that DARS applications can be made to NHS England to access the fruits of your labour, or is it still we can only access the mappings?
It's still only the mappings, I'm afraid, Jeff. We're still building the pipeline, and we're in the middle of reprocuring our research environment, and hopefully those two will tie up. Once that's done, if we're able to surface the OMOP model in our research environment, it'll be accessible to users of that. As to passing it back, I'm thinking, and because if you're still on the call, you might want to jump in here. It might be worth maybe there's some sort of collaboration so that our mappings could maybe feed into the work you're doing.
Yeah, I'm sure it's not the right thing to do to pass the transformed data back, but if you were to pass the mappings to the at one level, the data owner, i.e. NHS England, they should be able to run your mappings against their full national data set. And therefore create research accessible versions of the national cancer data in OMOP form. And that's a position I would love England and the rest of the UK to get to.
Absolutely. No, I think that would benefit everybody. And as I say, the mappings are free to take. Please make whatever use of them you wish. And excuse me.
So once again, I've put a general call out to the floor to ask if there are any questions or points that people would like to add. But if not, I might, Laura, just ask one other question. In one of your opening slides, you talk about EHR going through an ETL. Where should that ETL happen? Should that ETL happen at NHS England, or should it be happening at source, i.e. should it be happening within the cancer hospitals themselves who create national cancer data in OMOP form, or do you think that NHS England offers a form of data standardization and uniformity that is valuable?
I'm heading towards the latter. If the cancer hospitals had to do their own ETL, we might possibly get a richer data set, but of course it's going to be a lot of distributed work, and at least it might possibly eradicate a lot of their own quirks. But again, I can see the advantage of doing it at scale, and that we'll be able to get everything pretty much all at once, and presumably regular releases could be done. I'm just curious how the data is submitted. Is it taken from CDSs? Nicholas, are you still on the call?
So I know that we have to submit national cancer data sets in very specific NHS England data dictionary format, and then NHS England will merge and combine those. I have to say, I think I'm of the view that the right place to do the ETL is centrally, because we benefit from the uniformity of the data, and otherwise what you get is this concept of OMOP dialects, where one hospital's OMOP transformation doesn't quite speak to another form centres. However, NHS England at that point need to make that data accessible, and that's a completely different question, and one perhaps we won't be able to solve today. But I'm also aware, Antonelli, you have your hand up, so I'll come to you for a question.
Thank you. Thank you, Laura, for the presentation. I work on similar subjects. So I wanted to ask you which version of the OMOP Common Data Model you used, and if you used the episode and episode event tables that have been sort of suggested for oncology by the OMOP community. Thank you.
Okay, thank you very much. We use version 5.4. We didn't use episode and episode event because, if I remember rightly, some of the data information wasn't available, couldn't be safely inferred. We did actually use episode and episode event for mental health mapping, as that seemed to be a very close fit. But we didn't actually use episode and episode event for the cancer mapping. That might be something we could revisit in future. But we did find that it wasn't easy, possible to really safely build up the correct sequence of events because some of the key data points weren't present in NCRAS.
Yeah, no, thank you. I completely sympathize. The mapping in oncology is very laborious. So, yeah, thank you.
Brilliant, Laura. Thank you so much. In the interest of time and your voice, I'll move us on. But thank you again for your talk. So finally, before lunch, we're going to go north of the border and speak to friends and colleagues from Scotland who are going to talk to us about the mapping of the Scottish cancer data sets. So I think I've seen Carol briefly appear, and I also think, is Erin speaking with you, or are you speaking?
Yes, we're a double act.
Fabulous. So Laura, if you could stop sharing, and then over to Carol. Erin, the floor is yours.
Thank you very much. So, excuse me, Erin is going to share the slides so that she has control at the end of the presentation. So I'm just going to do a bit of scene setting, and then Erin's going to kind of dive into the more technical element, because I am definitely not going to claim to be an OMOP mapping specialist in any shape or form. So thank you very much for inviting us along to talk to you today. So just to kind of set the scene, next slide, Erin, is just to kind of tell you a bit about Public Health Scotland. And just to kind of say we are the public health agency in Scotland. Because this is what you want to do at the start of a global pandemic, we came to fruition on the 1st of April 2020. And we are a collaboration of kind of a couple of predecessor organisations. So our role is wide and varied in respect of public health, addressing public health priorities, vaccination schedules, and working towards population health framework in Scotland. But also we are the keeper of the national data sets in Scotland as well. So we kind of have multiple roles. And one of those is the cancer registry data set. So next slide, Erin. So I've, having listened to the previous presenters, that I kind of feel like I'm probably talking a bit to the converted here in respect of the wealth and breadth of data that's available. And we have that from kind of cradle to grave in Public Health Scotland, and we also are very lucky that we have a Community Health Index number, which allows us to do great linkage across those data sets. So that allows us to create linked OMOP Common Data Model data sets as well, which I think is a real strength for us. And one of the things that we have been looking at, and we've been working with HDR UK, was with their cohort discovery tool. So how could we make Scottish data available with that standardized layer that would allow researchers to go and see whether a research question is feasible or not. So we did a bit of work with them during COVID around kind of hospitalizations and COVID vaccination data. And then we've moved on to a second phase of the work, which has been looking at adding OMOP to hospitalization data, mental health inpatients, death records, and our cancer registration. So next slide, Adam. So part of that is work with the trusted research community across the UK. So within Scotland, we've collaborated very closely with we're the national safe haven within Public Health Scotland, but we also have regional safe havens in Aberdeen, Dundee, Glasgow, and in Lothian, which covers the southeast of Scotland. So this has been a true collaboration with our colleagues in Dundee and the Health Informatics Centre and the Regional Safe Haven, who've built up a lot of expertise and knowledge around OMOP. So we have been doing work with Erin and her colleagues around adding that, and then allowing us then to integrate our data across other study cohorts, national data sets, Genomics England you've just heard about, so that we can start to build that infrastructure across the UK that will allow people and researchers to come in and utilise the data effectively. So next slide. So yeah, as you've kind of heard, and probably will continue for a bit as we try to figure out how do we solve the wicked problem. So we still have lots of data in different silos. It is hard to find, it is hard to access. It's in different formats. Within Scotland alone, the cancer registry data is brought in from different sites across Scotland. So it's how do you create those national data sets that have a harmonisation across them, and then how do you make them harmonise with the rest of the UK, and then as we go further and further looking internationally as well. And that opens up the ability to do the federated analytics that have been mentioned so far, and looking at, you know, the provision of open and reusable code and methods and tools. So essentially, this is our first foray into that at a national level in Scotland, and it's been great to have the opportunity to work with our colleagues at HDR UK and on the Discovery tool to start that journey on looking at applying OMOP to our data sets, with a view that we can then start to feed that back in to Public Health Scotland's data sets for other uses, and then start to grow our kind of common data model across our data sets as well. Although interesting, your question there, Jeff, about where should you do that work, and that's a conversation that I've started having with colleagues back in National Services Scotland, who actually operate our kind of data warehouse and our data infrastructure, is the possibilities of work that's been developed elsewhere feeding back into the system to be able to service multiple purposes. So now what I'm going to do, and probably the bit you're all waiting for, is to hand over to Erin to talk about how she actually did the mapping of the cancer registry data.
Hi, thank you, Carol. So yeah, I think this was your bit. I think it's a snapshot of the HDR enhanced gateway where we have PHS metadata available and also available for querying on the cohort discovery. Yeah, so I'll just briefly explain how we transformed the data to OMOP CDM. We used a semi-automated pipeline, and the main reason we used it was not just to help ourselves, where we could replicate the process again and again, but also when other organizations come together, it's easier to share our mappings, and if they wish to use the same tools, they wouldn't have to do a lot of effort in transforming their data sets if they use the same workflow. So our process involved Carrot tools, which some of you may or may not have heard about. These tools are primarily used for mapping the data and transforming them to OMOP. And one of the key features of these tools is that you do not need the data to be at site for the transformation. So the mapping happens separately. You can share the mapping file and you can transform the data in a secure network at the data partners and where you have access to, or even if you don't have access to, someone else can run it for you. Here's the link to it if someone's interested to check out the tools. So yeah, the Public Health Scotland is responsible for collecting information of Scottish residents which are diagnosed with malignant or potentially non-malignant tumors. And we have a variety of information in this data set, demographics, tumor information, its diagnostic information, various stages of the tumor, other contributing factors, and the treatments that are involved during the whole process. This is just an overview of what the variable list is like under all these subcategories. So as you can see that there's a variety of information and a lot of variables involved, and not just the variables, but there are other data values that are inside these variables, which could be multiple. So it just adds to the complexity of the data set. We have used vocabularies from Athena to map the data. So we've just used these vocabularies. The good thing is that since we used the tools, we didn't have to download the vocabularies. These vocabularies are available on the Carrot Mapper tool. So you don't have to download or keep updating the vocabularies. They have this provision on the Mapper tool where they update the vocabularies regularly throughout the year. So you can refresh your mappings and make sure that they're up to date. So the first step, I'll just go step by step on how we transform the data and also the various challenges that we have encountered. Laura has explained it really well, or most of the challenges, but I'm also going to talk about the breadth of the data that we mapped. We transformed the demographics of all the patients in the cancer data set. One of the challenges was mapping the ethnicity, as there are, as ethnicity has always been an issue, what concepts to use and also which ethnicity to include. For now, we have only included the most recent recorded ethnicity of the participants in the data set. And also we have used the UK Biobank vocabulary for that. There are other vocabularies available like NHS, ethnic category, and race. And NHS ethnic category, although these have good concepts, but they are non-standard. So if anyone wants to use them, just be mindful that these are non-standard concepts. Because our core intention was to map the data to be available on the cohort discovery tool, the cohort discovery tool also has certain limitations. So we had to be very mindful where to put that information. So for now, the cohort discovery tool cannot access ethnicity from the person table. So we had to use the UK Biobank vocabulary to make it shift to the observation table, so it could be queryable by the researchers or other users of CDT. The next step was to map the primary tumor site and the morphology. In the dataset, we have the tumor site available and also the tumor type and behavior. In the code, the tumor differentiation is not available. So we don't have the last digit of the ICDO code available in the data. So we only mapped the ICD-10 site and combined it with the ICDO code with the behavior code, the cell type and the behavior code. Whilst mapping, you would see that there are certain codes that do not have an ICDO code. So for those, you cannot use the ICDO3 vocabulary to map those data values. So we mapped them just to the ICD-10, so that they were mapped to the SNOMED code instead of the ICDO3 code. So just make sure that when you're mapping the data where these codes are not available, you have an alternative way to support these tumor sites in your data when you're mapping to OMOP. Once we had mapped that, then we had moved to various tumor grades and stages. There were a lot of information available regarding that, and this took, I would say, the most time when we were mapping. I think one of the reasons was because the information was not available in an international code. All of these were local codes or codes that you need to just go to the vocabulary and find one by one where they could be available. We used combination of SNOMED CT and cancer modifier vocabulary to map these stages. During the mapping of stages, you have to make sure that you're mapping the stage concept ID, the cancer that it's referring to, and other numerical information that is linked to that grade or score. So for example, Gleason score, it'll have a grade and then a score as well. So you need to make sure that all this information is effectively captured in the table. So you have to use supporting fields like value as concept ID, value as number, to make sure that you have all the supplementary information and concepts available with the cancer site and morphology to make sure that it makes sense as a record. So yeah, this was quite a task. I cannot share all the concepts here in the presentation, but I have just shared snippets of what they look like. Then moving from the grades and stages, we tried to map the receptor status of different tumors, and we used the SNOMED CD vocabulary for that. It was relatively straightforward than mapping the grades and stages. We also mapped the site and the area of the tumor. Again, for the area, you have to make sure that you're capturing all the information that's available, because it could be multi-layered or multi-value information. So you would have a qualitative and a quantitative value associated with it. So you need to make sure that you're using the right concepts in the right column and the right domain for that. So yeah. After mapping that, we moved on to the cancer treatments. So there are various cancer treatments, surgery, radiation, chemotherapy, and then some detailed treatments were also available in the other variable. We tried to map them to our best understanding. Most of the concepts for these treatments were available in the SNOMED CT. there is another cancer vocabulary published by NNACCR. We have not used it, but it does have standard concept IDs available. So if anyone's mapping and they do find something in this vocabulary, it would be nice to know what they've used from this. We didn't get a chance because we were able to find it in other vocabularies, but also be mindful that this is no longer under maintenance. So they didn't have their refresh after September 2021. So there could be some concepts missing for that vocabulary. So overall, we mapped data from year 2000 to October 2024 of last year. Around 978,000 individuals were mapped, and we mapped about 1.26 million records from source. The mapping timeline we were very tight on time because it was a very short project and we had very limited funding available for it. So for the mapping, it took us about 12 to 14 weeks. We had to rush a few things, but so it was very focused work. We were quite focused just on mapping this because of the timelines, tight timelines. Also, I would say that most of the variables in the cancer registry were in the local coding scheme. So finding standard codes took a lot of effort and a lot of time. And our future plans are to share our mappings. This is one of the things that we've been discussing for quite some time. To document what we have mapped and also to share the mapping file with the other organizations to facilitate collaboration and knowledge sharing. Because there are so many people who are involved in this work, it would be helpful to avoid any duplication of effort if someone else is doing the same work that we have already done. And also, it would be nice for others to review what we have done and comment on it and validate our mappings. And so any kind of feedback would be really, really helpful if we want to run a refresh or redo some of the mappings that we have already done. So that would be really nice to do. So that's one of the things that we've been focusing on and talking about. So hopefully we get to do that soon. And the rest of you can see what we've done so far. These are some of the useful links that you can see for the mapping. And yeah, that's about it.
Brilliant. Aaron, thank you so much. Carol, thank you as well. It's fabulous to hear the work that you guys have done. There's an obvious first question, which I will jump in and take the opportunity to ask, which is, has there been any attempt to align the English cancer data work that's been done by Genome England and the Scottish data work? And in particular, has there been any thought of making the GitHub-like repository of mappings available to allow one-level third parties to map the homework of both organizations?
So no, but listening to Laura speaking earlier was going ding, 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 ding.
Yeah, I mean, at one level, it is an obvious idea, isn't it? That
yeah.
And at one level, speaking from experience, that there's the theoretical approach of comparing the mappings, which I have to say I find challenging and perhaps even unproductive. And then there's the very practical solution of trying to deliver a common project with a common aim. And if you were to set the challenge of running against the two data sets that we do have access to of what's the, I don't know, the age, gender, ethnicity and deprivation breakdown of triple negative breast cancers north and south of the border, what would that produce? And when we've tried to do that between Leeds and Bradford, we found that we've created different dialects that don't speak to each other. So I would desperately encourage a really simple, really high-level project that could compare those mappings, and that might be something we come back to in the very last session.
Yeah.
Are there any other comments or questions? Danny, welcome.
Yeah, Jeff, thank you so much. Yeah, welcome. Yes, I was going to say, this is, I think, your question and the answer you got demonstrates the potential and the effort needed to make OMOP really, you know, a game changer, right? So if we really manage to collaborate in a way that makes OMOP the normalized version of data across the UK, that would really enable collaboration across the country. So I'm not sure if there's anyone in the audience listening who has any kind of collaboration with the upcoming HDRS, but I think it's an obvious ask for the HDRS to facilitate these things happening, because there's no other way we're going to be ever able to run these analytics across different hospitals and nations within the UK other than normalizing data, making it interoperable, and then sharing analytics in a federated manner. So I think it's really great to see that people are in that mindset, and that there's an appetite to share, mapping code and so on, because I think we just need to make sure that we don't reinvent the wheel and we don't duplicate effort, right? I also know that there's someone in my team, Antonella, I think I saw her earlier, who is also mapping or has also mapped some cancer registry data, right? So I think we need to make sure that happens.
Brilliant. Any other questions or comments before we break for lunch? If not, thank you once again to all of the people who've spoken this morning. Really grateful for your contributions. Our plan now is to have a break. So it's 2:10. So may I suggest that we reconvene and start again? An afternoon when I'm delighted that we're going to talk about OMOP and cancer at 3 different levels. So we've got an opportunity here to speak OMOP Global, OMOP Europe, and OMOP UK. And I think personally, I think this is going to absolutely demonstrate the power of OMOP to do what becomes really interesting, which is collaboration above and beyond one's own local system. So first of all, I'm absolutely delighted to introduce Asiya Golazar, who is joining us, and on my agenda says that she's presenting from her role at Nemesis Health. But I have to say, I've known Asiya as the head of the Global OHDSI Working Group for Oncology. I don't know if that's a fair description of you, Asiya, but in many ways that was the context in which we invited her to come and speak to us. We in Leeds have been partaking in some of the studies that Asiya coordinates, so I'm delighted to give you the floor and give you the opportunity to describe that work and hopefully inspire other people to join your efforts.
Thank you so much, Jeff, and good afternoon, everyone. Really great pleasure to be here, and I see a lot of old friends online, so very nice to meet you all, and everybody else, very excited to tell you a little bit about what we have been doing and hopefully find collaboration opportunity. So as Jeff said, Asya Golazar, I am with a company called Nemesis Health, with which we do OMOP oncology at scale. But very specifically today and for this conversation, I'm going to talk to you on behalf of the oncology work group. I've been leading the oncology work group for a long while now. I'm losing the timing. So just telling you a little bit about the efforts that have been going, what we have accomplished. Also very excited, interested to hear from your experiences, the hurdles, the problems, or any potential collaboration opportunity, because we really want to make advance the oncology capability within OMOP, and we need you for that. So I hope you can see my slides, and I'm going to start. Just going to talk a little bit about the state of affairs. So there's a lot happening, and we are all here to do oncology research. And we are here because we all understand that as Jeff said, we have to work together. We have to come together, learn from each other to do it more and to do it better. The current state of affairs when it comes to RWE in oncology, thinking about everything that is happening, if you open PubMed and search RWE, real-world evidence studies in oncology, is that we're still lagging behind in terms of the quality. This is just an assessment that was done and a publication, a review publication that was published last year, talking about the need for quality, the need for international collaboration, promotion using standards to be able to generate reliable and relevant evidence for oncology that can inform guideline and practice, basically why we are here. A lot of reasons for that. I'm definitely preaching to the choir, but just to bring everyone on the same page, a part of it is the disease itself. Cancer, big, a lot of number of people with cancer. The moment we look into specific cancers, indications, looking at specific treatments, we're looking at small counts. This is just an example. A recent study that we've done, that Jeff and some of the other colleagues on the call were also a part of that, was looking into bladder cancer with over 600,000 new cases annually diagnosed. When we look into the metastatic patients, the proportions that are treated, and looking at different treatment groups or categories that the patient can receive, all of a sudden we see these like tiny pieces of pies that you can't basically see. That's a problem. 0.7% of the total bladder cancer patients, if they're eligible for a treatment within your institution, you're going to end up with a double digit or something that doesn't allow any of us to make meaningful evidence. We are here because we know that we want to make it together, combining and hoping that these little pieces of pies can come together to make it bigger and meaningful. The other problem is the details that we need to do for cancer. This is just an example of a drug label, Tabrecta, for treatment of metastatic non-small cell lung cancer for patients with MET exon 14 skipping mutation. There are too many details for us to be able, that we need to know to identify the patients. So it's not like tuberculosis, that we just need to know the patient has tuberculosis. We need to know there is a tumor with specific histology, something in the lung with a histology of non-small cell lung cancer, non-small cell, that is metastasized. Also, these patients have to have specific mutations. So the details are issues because we need them. They're not always available within like the data, or they need to be pulled together. Creates another problem. The data are fragmented. Some of the data are in the registry, the genomic data can be everywhere and nowhere. That's the experience that they're scattered all around in PDFs, VCF, ingrown tests, the homegrown test, or the tests that are coming from vendors. We have pathology reports from imaging reports. They're all fragmented, and sometimes it's hard to identify where they are. They need to be linked together. We also sometimes have problems that patients move from institution to institution. They don't carry their history and the information. So we lose the continuity of the patient care. Okay, sorry. And then the quality. We're dealing with quality issues. We know that there's a huge call. These are calls like coming from ASCO and ESMO that we have to do something about the quality of the data and the depth of the data to be able to generate meaningful evidence. So what have been doing? So the oncology work group has been there since 2017-16 with one goal, enabling observational cancer research. Similar to what everybody else is doing using OMOP, we wanted to enable same approach for oncology. So this is just a reminder for everyone that and we can get into the details later. We have 3 subgroups within the oncology work group: the outreach and research, which specifically focuses on research, and any potential opportunities for collaboration or grants that can enable a lot of the work that we do. We have 2 other subgroups, one focused on genomics, on omics in general and cancer, and then the vocabulary and development subgroup that is tasked for maintaining and developing vocabularies or specific tools that would help ETL or for future analysis that are specific to oncology. And over the years, we've developed the OMOP cancer model, which has 3 specific modules: the way we represent the disease, a combination of conditions, which is an ICDO representation, and a series of diagnostics attribute, what we call diagnostic modifiers, and we represent with cancer modifier OMOP genomic vocabulary. We've introduced the cancer treatment model, which allows us to describe cancer treatment more than what's currently available at an individual level treatment, to describe the regimens, cycles, and line of treatment. And finally, the episode, which you can actually, that's the addition to the CDM that was introduced a few years back for oncology first specific, and is being used. It allows you to describe periods of the disease that have specific meanings: a progression episode, a remission episode, a treatment, first line of treatment. As a reminder, these are the vocabularies that we use and recommended for different representation of different parts of cancer oncology OMOP. And as a work group, as a community, having the model together, we have to work on 3 different pieces to make this work and to enable high quality RWE. First is the data and the data quality, then coming together and setting up a network so we can deal with the number problem, and then cranking out evidence, setting up the foundation and do that. So how do we do with the data? We know that there's a lot happening and there are a lot of details that we need to know, and all of them are important. There's no need for that, but we need to prioritize to make it work, what to do first, what next, and so on. So we take the information to do the prioritization from the community, from everyone who's sitting here and everyone who's within the OHDSI community. Going through the use cases, so these are just examples, when the ones that you see in the table on the left, of different use cases that have been submitted by different community members. And the use cases help us identify what data elements we need and then use it for prioritization. We have an ongoing assessment of the use case, a use case repository that we update and assess biannually, that helps us identify what it is that we need to focus. So for example, pretty much in most cases, we need a proper diagnosis of the disease, meaning we need to have histology plus topography of the condition. We have to make it work from vocabulary representation, mapping, and then finding where these resources are and whether there's help needed in bringing them together. Sometimes histology is in one resource and topology is in another. How do we combine them? So creating these capabilities within the workgroup. Stage metastases, we keep updating these lists based on what we are developing and based on the use case. So this is a big call for everyone who hasn't seen that This has a link, and I can send a link to the chat. Please submit your use cases. That would help us make advancement and help you get to your use case needs sooner. Once we identify that, we do, as I said, use case driven prioritization. We know what's needed, what type of vocabulary needed, and we work with the data partners or the network partners in terms of their readiness, the quality of the data to do the oncology study. And once we identify the issues, we work on solutions or fixes that can, a part of it are the requirement of an update in the vocabulary or fixes in the ETL or providing the mapping or helping the data partners in bringing the quality to what is needed. The data quality assessment that we've implemented, which we call it the oncology data readiness, is something that we've worked with in the work group a year and a half ago. We started this work and we're expanding every day, which basically goes within the data and look at any cancer-specific information. Only at the concept record, we don't touch patient-level information. The query just gives all that information back, and through that, we can assess the quality of the mapping and the depth of the data and help us create a report for the data partner to tell them what they have, how good or how bad the quality is, and what are the issues that they need to fix. Data partners also would get a short-term or a long-term fix. We iterate this process until all the issues are fixed. So all the network partners, every data partners that are within the oncology, OHDSI oncology network, have the same quality so they can answer the questions. This is basically an self-service. All the scripts are available online at the OHDSI, oncology.ohdsi.org. And yeah, if there's any data partner here who would like to check the quality of their data and see where they are and understand like how they can be improved, happy to do that. You can either ping me later or go to the website and use the self-service. So when we started, this is just an example of what we generally produce, just we look at both standards and source mapping. We identify issues within the databases, and we give a very, very detailed report to each of these data partners. And then we work with them to fix the issues with the hope that we get to these pies, like what we see here for Leeds University, of no issues. If all of our data partners get to this clean situation, then we have full confidence that the evidence that we're generating is correct. We have the right data at the high quality. This is just an initial through the iteration, and when we do the studies, pretty much all of our data partner will come like all clean, so we can start the work on evidence generation. And as a part of the fix, we're also regularly releasing new vocabularies or changes or updates to the cancer vocabulary. As an instance, in July, we release an off-cycle update to the cancer vocabulary with these specific changes that we identified. All of these were identified through this oncology readiness assessment, because at that point, we realized what's needed for the use case and where are the gaps that we can address. This is again available on the website. We are working with the vocabulary work group to integrate it as a part of the OHDSI vocabularies. And then once the data are good, we can develop a network. We can come together and do evidence generation. So far, through the studies that we've done, and Jeff also mentioned, we've done a couple of studies, and we started with one study and one question, improving the data and generating evidence. And at some point, all the network partners were more interested to do more. You've already put the work in. You've cleaned the data. It's good quality is good. You've shown that you can generate evidence. Why stop? Let's crank out more evidence. These are all actually the majority of the data partners that we are working with at the moment within the oncology work group, and under an umbrella name of FALCON, or Federated Alliance for Large Cancer Oncology Network. And then we can do evidence. So two examples of the two large-scale studies that we've conducted this year was a study on metastatic non-small cell lung cancer that was sponsored by ICANN Precision Medicine in Finland, with the goal of exploring the real-world treatment landscape of metastatic non-small cell lung cancer. When I first heard the title, I thought, oh, like checkpoint inhibitors were introduced in 2015. Why are we asking this question again? Well, I didn't know much about the difference in reimbursement pattern back then and how important it is to actually describe the heterogeneity. Went back to the literature. There's a lot of publication on the uptake of immune checkpoint inhibitors on metastatic non-small cell lung cancer, but there is not much about the geographic heterogeneity of the uptake. What we can learn about that, from that, and its potential impact, the potential impact of the introduction of these treatments on survival and on outcomes, and how this can be potentially used for reimbursement negotiation. We started the discussion end of last year, and end of March, within three and a half months, we had a network of 22 data partners with high quality data. So basically everyone went through the tireless work of checking the data, working on the improvement of the quality. So at the end of March, we had 22 data partners with high quality data that they could generate evidence, we could review, and we have presented some of the results in ESMO 2 or 3 weeks ago. Earlier this year, the other work that we started was to look into guidelines. It's what was called guideline-driven evidence, and what we're calling Guideline-a-thon. That was a collaboration with the European Association of Urology throughout the discussion that we are having with them in developing a framework for integrating RWE into guidelines. So instead of each of us coming and trying to ask a question, why not turning the guidelines into electronic decision tree that can be phenotyped to become computable, and then we can have like a systematic framework for asking all the different questions that guidelines can ask. So I'm going to give you a little bit of snapshot of both of them. As I said, these are all happening under FALCON, or Federated Alliance for Large-Scale Cancer Observational Network. FALCON brings it all together. FALCON is a network of high-quality data partners, and everybody is extremely well equipped to generate evidence. We have a lot of good examples of people who have been great partners in this journey, in this meeting. So FALCON Lung, 22 data partners, 9 countries. At the time of presentation, 33,000 non-small cell lung cancer patients from the countries, like extremely geographically diverse. These are some snapshots. All the results are available in this Shiny app here that you can explore. We have ever since gathered more data partners, and the total number of non-small cell lung cancer is right now more than 100,000. One of pretty much probably the biggest and most diverse study on non-small cell lung cancer in a network. So when we started with bladder, we started thinking about how we can inform guidelines. Guidelines come from a need, patients requiring treatment. Guideline developers sit together, put the studies, look in the study assessment, is it eligible or not? Yes, there is a treatment recommendation. If you look into the whole process, one thing is missing, a feedback loop that can tell us whether these recommendations are relevant, are really these recommendations adhered to, and how generalizable are these recommendations. Should they all be on patients who have ECOG 1 or 2, or if patients have like ECOG 2 or below 2, or if they have ECOG 2 or 3, they can still benefit. To what extent can they benefit? What about older populations? Are there unmet needs? All of that can potentially be done, and also generation of de novo evidence with RWE. We can generate a richer corpus of evidence. So this is an example. This is the European Association of Urology 2024 guideline for the treatment of metastatic bladder cancer. So the entire guideline can be turned into a decision tree like that. And theoretically, all of these nodes can be turned into concept sets and phenotypes. Once we have that, we can answer a lot of questions in a one shot, not generating multiple studies to answer the different questions in different times. That's the general approach. So what question could be asked? Enfortumab plus Pembro was approved in 2020, end of 2023 by the FDA and mid 2024 by the EMA for the treatment of metastatic bladder cancer patients, first line of treatment. The guideline itself says that whatever we know comes from one trial. We actually don't know who else is eligible or not. There is an opportunity here. We can do it. That's what Falcon Bladder is also trying to do that. And if you look at the network, this is just 22 sites that I could basically show the results like 2 weeks ago. So our biggest tertiary care in the US at that time, using the eligibility criteria of de novo metastatic bladder cancer, they only had 20 patients. We can't really do much with 20 patients. Looking across the network, we have some counts here and there, but none of our databases is alone by themselves can generate any meaningful evidence. And we are still at this situation that the drug is not reimbursed everywhere, so we know that this picture will change. But for a timely question like that, none of us can do it unless we come together, and that's what Falcon brought. So as of beginning of October, the network, in terms of the sample, has more patients than just the clinical trials, and we can start looking at a lot of questions that are still unanswered. This can be done for a lot of others, so obviously the counts and everything else I'm not going to show you for other treatment options, and it's much higher, but that's what we're trying to accomplish. And this is basically the hard work of every single 45 data partners that we have within Falcon Bladder, of putting the data there, making sure the quality are there, and working together to generate the evidence. So is it possible? Yes. Can we do it together? Yes, but we have to work on the quality of the data and come together as a federated network to make it happen. I would like to invite you all, and if there is an interest for any of this, we need your help. None of this is possible by just a few of us. What we built with Falcon is really the hard work of every single data partner and every single group, which I can't be thankful enough. And if we don't do it together, we won't have success. So please consider joining us if there is anything, even if you want to complain that something doesn't work. We're here to help, and we're here to work together to make it better. And thanks again for having me.
Azir, thank you so much. I send you a virtual round of applause. And at one level, the thanks for the work that you've done over a period of, goodness me, 8 years or something, leading OHDSI's direction in cancer, but then also, more importantly, the delivery of the Falcon network. And then I think the study that you've done most recently, well, you're doing most recently on the assessment of the bladder cancer guidelines is absolutely a vision of the future. So we're constantly asked to judge our performance against published guidelines, and the idea that we can turn those into a computational process that just runs against our data and scores each center against the guidelines is a vision of the future. And I hope that's something that becomes much more widespread and established at a global, European and national level. So with those few comments, once again, I'd invite anybody else to raise questions or comments or thoughts or challenges for us here. Azir, if there are centres here today who are interested in joining your network, how long do you think it takes for one of those centres to begin to usefully and actively contribute data to your network?
So That's a very, very good question. A lot of it is basically depends on how fast we can work together and help you. But if there is a center, so the process can be done pretty fast. And that was what we accomplished through Falcon Long. Within 3 months, we had 22 data partners with high quality data. So, and that's like for some centers, it was like a couple of rounds of assessment and trying to through troubleshooting or figuring out what the issues are and doing the work. Some centers like yours, it was already great. So it was much easier, but it's usually like within like given the back and forth and just not thinking about, so because sometimes IRB approval would take some time, but in terms of improving the quality is quite fast. We can turn it around in 2 or 3 weeks if we do the proper back and forth. That's very nice. What we're trying to do with the oncology readiness assessment is to add other details. So we're expanding the capability of the quality assessment. For example, we've added labs to what we're doing because we know a lot of these treatment decision making are based on the function, like the different organ functions, and whether like the kidney is okay or the liver is okay. So that creates like a little bit more deeper work. But we're also learning quite a lot from the data partners. What kind of data are available, how they're mapped, or if they're not mapped, why? So we can have very, very targeted approach for improving the depth and like improving what's basically might not be even available in OMOP.
Right. I guess my other question is it possible? So one of the things that intimidated leads when we first started on this journey was the depth of the OMOP hole, and you can dive in quite deep. And the idea of doing highly detailed studies based on molecular markers and targeted therapy to examine third-line therapies and recurrence and progression events is quite deep down the OMOP hole. Whereas actually there are quite high-level studies that allow people to dip a toe in the water, if you like. And at one level, one of your first slides talked about, how many patients do you have diagnosed with bladder cancer? Of those patients, how many had metastatic bladder cancer? Of those patients, how many had chemotherapy? Are really quite high-level concepts that you don't need to do a full mapping of 200 data systems within your organization to do. So I think it'd be really good if we can get across the concept that any contribution to the Falcon network is useful. You don't have to deliver the full study. So some of the questions might be possible to be answered in 50 centers. There may only be 10 centers that can go to the full depth of the study.
Exactly.
So I think that would be a really good principle to establish that there are simple, moderate, and then complex questions which are being asked, and contributions are equally valuable at any level.
Absolutely. And that was basically, so a couple of things. When we started doing the oncology, there are a lot of questions, right? And then none of us, even if we have all those data in our system, we will never get to that perfect level that we have everything in place to answer all the detailed question. And that's why we implemented the whole system of let's take a look at the use cases. Let's see what the use cases need, and then let's do it step by step. And then we can generate evidence. We can't wait for 10 years, for 15 years to have everything ready and say, hey, now we can crank evidence, because by that time, it's going to be like the train has already gone, right? So we need to start showing the value, and then using that to communicate with the clinical world or the clinical community what it is that we can bring, and then build as we are delivering. So as you said, with the Falcon Long, non-small cell lung cancer, not everybody is there yet in terms of having their tumor registry, for example, linked, or have like very detailed information in their histology to correctly identify that. But 22 data partners have it. So they worked on the quality of the data, and we generated evidence. With bladder, there's a lot that we can do without histology, as long as we have others. Let's create that base. After that, there's already a lot of questions of what's happening to FGFR to FGFR3 fusion. What's like, who's using that? What type of molecular testing that are happening? We will do that when we create a high quality base. So this is exactly, everybody can do some oncology research, but we all have to come together and so then go deep where it's possible and show the depth or the width in the other places.
Great. Thomas, can I come to you for a question?
Yes. Thank you very much, Asia. It was a delight to work with you on the Falcon side of things. I think one of the things that we found was that like the differences in the mapping that's been done between different data partners and the complexity in the oncology model. I think kind of the way I see it is that as more people start to map the oncology data, we're going to start to see different types of or different people mapping that data that aren't necessarily part of, say, the Falcon network or aren't necessarily part of the OHDSI oncology working group. So what I was kind of wondering was, like, do you foresee us to be in a place where we can routinely run sort of the readiness assessments? If we know that we're running, if we know that we're going to be doing oncology data, this is what needs to be done in order to map it to the correct places, and that it's also kind of updated on an ongoing basis as we develop the model moving forwards. Because I think that's something that was highlighted kind of in the morning as well, is that even within the NHS side of things and from our Roche perspective is that we do see differences in just terminology that's been mapped or that we've mapped it into one place or another. And it's just good to highlight those things up front rather than necessarily finding it when we actually add data to the network itself.
No, absolutely. That's our goal, to create a whole like automatic self-service. Right now, I'm having a lot of fun because I get to meet people like you and then collaborate together. But the goal is to have it automatic, so we actually everybody can make sure they are on the most updated according to the conventions and vocabulary. And also that creates a feedback loop for us, because you guys have different source data, you have different needs, and we don't know. We've created a model that constantly needs to be updated. We need to make sure whether like the mappings are still correct, so whether it adheres to the conventions and all of that. So to create that feedback system for the work that the oncology work group do, making sure that everything is updated, creating also proper documentation for that, and to make it easier for all data partners to do it very fast. So, and this is already working, but just needs to be tested and refined. So the goal is that you upload it and then you get a report quite fast, like in a couple of minutes. You can assess it, you can ask questions, but that's the general goal. Make it easy for everyone, but make sure that everyone has to have the high quality. I've done a lot of these work, and what's quite frustrating and what kills the studies are the different ways of mapping. So you either have to go through the entire vocabulary system to find all potential ways that metastases can be mapped. That's killing, because you can't do that, or sometimes like a broken convention. And I know a lot of them also is on us as a work group to make it easier and provide the documentation and keep it updated. So we're doing it step by step. So we know the pain, let's fix it. We are also initiating a whole new effort on documentation, because our documentation needs to be updated, but we want to work with the community to do that. So people tell us what doesn't work, we fix it, we release it, we move to the next chapter. There's a lot happening, and we need all of you. So we've learned a lot through the work that you've done, Thomas, in your group, and I'm sure there's a lot more people here that can help us.
Brilliant. Azir, can I move us on in the interest of time? Thank you so much again for making time to speak to us today. But coming slightly closer to home in a number of ways, can I introduce Stelios, who is going to talk a little bit about the work that has been going on with Cancer OMOP within a European collaboration. So Stelios, over to you. The floor is yours.
Thanks for the introduction, Jeff. And good afternoon, everyone. My name is Stelios Stefanos, and I'm a real-world data analyst at Leeds Teaching Hospitals NHS Trust. I joined the team here at Leeds around 2.5 years ago, and since then I've been leading the development and maintenance of our OMOP database. And the overall goal of my work is to make the Leeds data more usable, more standardized, and also more connected to research networks across the world. So today I'll be speaking about some of the cancer-specific OMOP research that we've been working on in Leeds, specifically within the DG1 pilot network. So firstly, I will describe what data is available in the Leeds OMOP database, and then I will explain what DG1 and MEDOC are. Then I will dive a little bit deeper into the 3 studies that we contributed to as part of the DG1 pilot network, focusing on COVID-19, on metastatic non-small cell lung cancer, and also on metastatic breast cancer. We started building, as I mentioned before, the Leeds OMOP database in 2023, and it's still being actively maintained and extended as we participate in more research studies. It currently includes data from about 1.5 million patients that have a diagnosis in the trust, and more specifically around 100,000 patients with a cancer diagnosis from 2003 to October 2025. For every patient that has not opted out of their data being used for research, we have diagnosis codes, inpatient admissions, surgeries that were delivered, as well as antibiotics that were prescribed. For patients with a cancer diagnosis, we actually have much more detailed information, including staging, morphology, some biomarker data, treatments, and outcomes. And you can find more information on the Leeds OMOP database in the HDRA UK gateway, and the QR code is at the bottom of the slide that you can scan. So developing this database essentially enabled us to contribute to large-scale OMOP studies on the national level within the HERON network, which Ellen will describe in more detail after my talk, at the European level through DG1, and finally at the global level through the Falcon network that Asya has just talked about. And as I mentioned previously, I'll mainly be focusing on the work we have conducted as part of the DG1 network that has been established by DigiCore. DigiCore is an international consortium that aims to digitize cancer outcomes research across Europe. And to do this, DigiCore aims to bring cancer hospitals and industry partners together in order to carry out research within the real-world evidence space. And this allows partners to collaborate and share knowledge more effectively, of course, with the ultimate aim of improving cancer outcomes across Europe. As a first step to achieving this, DigiCore established the DG1 pilot network, this of 6 digitally mature hospitals in Europe. The aim here was to enable these centers to achieve interoperability and high data quality using the OMOP Common Data Model. And through this network, we can now link molecular and clinical data and then carry out fully federated analysis in order to answer some important clinical research questions. And ultimately, by learning from this pilot network, the overall goal is to help scale up other less digitally mature hospitals in Europe. And this is currently being done in the DG1i3 project. At the beginning of DG1, we quickly realized that we don't only need a common data model, but we also need a common clinical language between centers. Cancer, as we all know, is a very complex set of diseases, and clinical practice and the way that data is recorded and coded in source systems can differ massively across centers. So this led to the development of the MEDOC dataset, the minimal essential description of cancer dataset. MEDOC was developed within the network and by the network to identify the minimal set of data items that each center could reliably provide and map to OMOP in order to be able to carry out multi-center observational research. The 38 MEDOC data items cover core domains, including demographics, diagnostic information, for example, cancer site, morphology, and the date of diagnosis, key treatments, as well as outcomes and follow-up. And this also comes with some guidance for standardized coding and vocabulary mapping. While this minimal set doesn't capture every nuance of oncology care, it maximizes participation and comparability across centers. And if you're interested in, or if you're in the process of mapping your oncology data to OMOP, I'd encourage you to read the MEDOC implementation guide paper that was recently published in the ESMO Real World Data journal. The guide offers a lot of information on which data items were mapped to for DigiOne and how we managed to harmonize these.
Stood beside Sachiko, gripping the
To give you a sense of how we harmonized clinical concepts across centers, this figure shows the MEDOC diagnosis code mapping tree. The goal here was to take the wide variation in how tumor sites and morphologies are coded locally, and then map them into a consistent structure aligned with the OMOP vocabularies. This structure helps resolve mainly two problems that we see across different centers. First, the inconsistent use of coding systems, and second, differences in the granularity of the available data. So using this tree across DigiOne means that any cancer diagnosis will land in OMOP in a more or less standardized manner, regardless of what the original source system stored. The second example is how we standardize the data of diagnosis. And this is a deceptively simple data item, but in reality, centers capture it slightly differently. For example, some centers record the date of the pathology report, some the date of the first clinical suspicion, some the date of that the tumor board confirmed the case, and others have multiple.
Stelios, you've just jumped onto mute. Try again. Yeah, you've come back.
Awesome. So MEDOC defines clear rules on how to record the date of diagnosis in OMOP. And as you can see in this table, that outlines the hierarchy of potential dates, so from pathology to imaging to clinical documentation, and then specifies which one to use if several.
Stelios, it's happened again.
It does say that I have been muted, so I don't know what's happening.
Keep going, Stelios. We'll try not to mute you.
Yeah. So essentially, harmonizing the date of diagnosis matters because a lot of the downstream analyses depend on a consistent definition of diagnosis dates or on a date of diagnosis. So this could be, for example, survival analysis, time to treatment metrics, or cohort entry logic. By using the same prioritization rules across DigiOne, we can minimize ambiguity and also ensure that all centers anchor patients' timelines in the same way. Okay, so once centers in the DigiOne network had converted their MEDOC data items to OMOP, it was time to design and carry out some research. The first DigiOne study that we carried out investigated the impact of the COVID-19 lockdowns on new cancer diagnosis and 12-month survival across 5 European cancer hospitals, covering a period before the pandemic, during the peak lockdowns, and then the subsequent recovery phases. And this was deliberately designed to be a very simple study using only 5 data items, with the aim of proving that multi-center research using OMOP can be done at the European level without sharing individual level patient data. 5 of the 6 centers participated, and we analyzed data from 124,000 patients in this study. On the graph here, we can see the decrease in the volume of primary cancers diagnosed each month from baseline to hard lockdown, and this was statistically significant for hospitals 2 and 4. At the end of 2021, we see a trend back towards the baseline for most centers. This graph shows the relative one-year survival of patients diagnosed each month during and after the pandemic compared to the baseline, so before the pandemic. And here it's interesting to see that UK was affected the most out of all centers, being Hospital 2, with a significant drop in survival for patients diagnosed in the first half of 2020. The second study within DIGI1 focused on metastatic non-small cell lung cancer. And for this study, we had a study-a-thon in Lisbon in March 2024, where researchers and clinicians from the participating centers got together to work on this project. Overall, we analyzed data from 1,294 patients, 73% of which were first diagnosed at the metastatic stage. During the study-a-thon, we were also able to plot fully federated Kaplan-Meier survival curves, which you can see here, using the Vantage 6 federated learning platform. And these curves show the overall survival of patients across the entirety of the network rather than on a center-by-center basis. I think this is particularly important because we wanted to stratify patients to subgroups based on their metastasis location. And some of these subgroups were rather small in individual centers, and we wouldn't be able to get these curves without a fully federated approach. Here, it's important to highlight that over half of the study cohort were found to have metastasized to more than one location, and their median overall survival was comparatively shorter than other subgroups. The lung and pleural metastatic subgroup had the best prognosis out of all. And finally, we saw that there is statistically no significant difference in the survival of patients with multiple metastatic locations with and without bone metastasis. Another working group in the study-a-thon focused on developing a line of therapy algorithm, which was then validated using the data from the OMOP databases in the participating centers. The plot here shows the proportion of patients having a specific treatment as a line of therapy 1 on the left, and which treatment they go on to have as line of therapy 2 on the right. And developing these algorithms is crucial for guiding treatment decisions in a consistent and informed way, and ultimately improving care for patients with metastatic non-small cell lung cancer. Finally, we have the metastatic breast cancer study, which is still ongoing. 5 of the 6 DIGI1 pilot centers are participating in this study, and we're looking at patients with a diagnosis of hormone receptor positive, HER2 negative metastatic breast cancer between January 2018 and December 2023. Besides the main cohort of patients, we also defined subgroups based on their disease status. So we have the de novo metastasis group and the recurrent metastasis group. Each of these subgroups were then divided further based on the HER2 status. So we have groups for HER2 low and HER2 zero. Then we analyzed variety of MEDOC data items spanning from demographic and clinical patient characteristics to more complex data items like biomarkers, biopsy and rebiopsy status, and the treatment that was delivered. For the analysis, we extracted a study-specific dataset from each OMOP database as a CSV file, and then this CSV file was linked to a Vantage 6 node ready for the federated analysis. So next, I will present some of the preliminary results from the first round of analysis, which will be presented at the San Antonio Breast Cancer Symposium. So overall, we analyzed data from 551 patients that had deep phenotypic data, a third of which were de novo metastatic, and 2/3 had a relapse from a previous early stage breast cancer, with the de novo group being slightly older in age than the recurrent metastasis group. This plot shows the HER2 status of patients that had a relapse from a previous early stage breast cancer, and more specifically, if the HER2 status has changed between the initial breast cancer diagnosis and the metastasis diagnosis. Here it's important to highlight that almost 80% of the patients who relapsed have had a rebiopsy at relapse. And this small part here, about 6% of patients were actually HER2 positive at the early stage and then became HER2 negative at the relapse stage. And this highlights the need for rebiopsy at this stage, as the HER2 status typically guides clinical decisions regarding treatment. In addition, the results from this preliminary analysis show that more than half of the patients could benefit from specific therapy targeting an actionable gene mutation. In terms of the next steps for this study, we're planning on developing a line of therapy algorithm, as well as carrying out federated regression analysis to explore some important clinical questions. Specifically, we want to analyze the available data to answer questions like, after CDK4 inhibitors as the first-line treatment, what is more effective as second line? Continuation of endocrine therapy or switching to chemotherapy? And are CDK4 inhibitors in the first-line setting less effective in patients with a BRCA1, BRCA2, or PALB2 mutation? And hopefully by answering this type of questions, we can generate real-world evidence that can support the personalization of treatment for future patients. And this brings us to the end of my presentation. I hope this gave you a clear sense of the kind of research that we can drive at the European scale using the OMOP Common Data Model. Thank you.
Stelios, thank you very much for your tour de force around European OMOP and cancer. Are there any other questions and comments before we move on? I get to ask questions of Stelios every Monday morning, so in the absence of other questions, Stelios, forgive me if I move us on in the interest of time. And Ellen, are you with us?
Yes, sir. Yeah, I am.
So Ellen, hi, thank you for joining us. And Ellen's going to talk to us about the UK OMOP network that Danny and colleagues from Oxford lead, and the works that they've been doing, and in particular, how those studies may begin to look at cancer. So Ellen, the floor is yours.
Am I sharing the slides okay?
Yeah, that's perfect.
Perfect. So yeah, today I'll be presenting one of the studies that we've done with the Heron UK network, which is on the use of high-cost antineoplastic agents in English NHS hospitals. So this is actually part of a wider study that looks at a lot of different high-cost medicine, but for the sake of this presentation, I've just pulled out the results that are relevant to cancer. So I'll just go a bit about me. My name is Elian Rolands, and I work as a research assistant in health data sciences at Oxford as part of Danny's team. And I primarily work on Heron UK stuff. This study in particular was primarily the work of Ed Byrne, but unfortunately he couldn't actually make it today. So firstly, a bit of background. Antineoplastic agents, also sometimes referred to as anti-cancer drugs, are medications that specifically target cancer cells. There's a large variety of them, including alkylating agents, PKIs or protein kinase inhibitors, and monoclonal antibodies. So these drugs can be very expensive, especially if they are new drugs or designed to treat rarer forms of cancer. And this is obviously because of things like cost of research and development, and also in the case of rarer cancers, just lack of price competition. So in general, high-cost drugs make up a large proportion of overall healthcare costs. Just as an example,
UCLH, approximately 70% of total drug expenditure was on high-cost medicines. However, we don't really know much about how these drugs are actually being used in the real world and who they're being given to and why. There's also some issues in terms of access to high-cost medicines because they're usually driven by things like geography, socioeconomic background, and systemic factors. So, you know, if, for example, if you have a rare cancer, you might have to go to a specialist centre, which are often going to be concentrated in large urban areas, which in the UK basically just means London. And so it becomes a bit of a postcode lottery, really, whether you're able to access the treatment easily. Obviously, travel is an option for some patients, but this can be very expensive, require time off work, and also can be quite difficult to manage, especially if you're in the later stages of cancer, maybe traveling just isn't feasible. Also, within the UK, there are differences between different NHS trusts, especially with funding, which could also contribute to unequal access to these treatments. So, as I kind of briefly mentioned previously, this is a study done with the Health Data Research OMOP Network, or HERON, which was established last year, I think, initially, and it's in collaboration between HDRA UK and our team at Oxford, and currently has 7 data partners, 5 of which are hospitals and 2 of which are community care databases. So for this study in particular, we included 4 of these data partners, which are all hospitals. 3 are based in London, so that's Barts, Gosh, which is a paediatric hospital, and UCLH. And then we also had Lancashire, which is based in the north of England. We identified high-cost antineoplastic drugs by using the NHS England high-cost drugs commissioning list. So in total, this list has about 800 drugs, about 150 of which are antineoplastic agents. And we did a cohort study with the aim of describing which high-cost antineoplastic agents were used in these hospitals, who received them and why. So we did this by getting counts of new users of these drugs from the 1st of January 2022 to the end of database, summarising characteristics of new users, things like their age and sex, and identifying conditions of interest associated with their use. So just to clarify, the January 2022 start date is because this was the start of usable data, or when electronic prescription records were available for these hospitals. And the conditions of interest we identified through like a data-driven large-scale characteristics. And then we did the study code, and we used a lot of the packages that we make and maintain in Oxford, so things like code characteristics, code constructor, and these are a mix of OHDSI and Darwin EU packages. So some results, so obviously because we've only got 150 high-cost antineoplastic agents on the list, I've reduced it down to just the ones that we see most commonly in these hospitals, and so these are the 5 most common ones we see. So the first thing to point out really is that there is some consistency in that rituximab and sirolimus, sorry about the pronunciation if it's wrong, but these are seen in all 4, or at least the majority of them. And also something else to point out is that UCLH has the highest number of new users for these drugs, whereas Lancashire has a fairly low number, only looking at 15 new users for rituximab in Lancashire compared to 671 in UCLH. And another thing to note is that there is a lot of overlap, specifically between Barts and Lancashire. However, for Gosh and UCLH, we technically see that they have more unique drugs that you don't really see in those hospitals. So here are some of the outcomes of interest that we identify. So these are specifically cancers or cancer-related conditions associated with the use of these anti-cancer drugs, which are recorded in the 2 weeks before and after they started treatment. So along the bottom, we have the antineoplastic agents of interest, and then on the y-axis, we have the proportion of new users with a record of those conditions. So I'll just explain quickly why we don't see all 5 of the top drugs for each of the data partners, basically just because for some of these drugs, there aren't any cancer records within 2 weeks either side. I think the main reason for this probably is that some of these drugs might be used for more advanced stages of cancer, which would maybe explain why we're not actually seeing records of cancer around the time that they're being used. But for the drugs we do have results for, we mostly see leukemia as a condition of interest. So for Barts, Gosh, and Lancashire, it's all kind of different forms of leukemia, whereas UCLH has a lot more variety. So some of them is leukemia again, but also things like multiple myeloma and lymphomas as well. And then they also look at that median age of new users. So again, we have median age along the bottom there, and then the drugs of interest on the y-axis. So the results for Gosh, because it's a paediatric hospital, is as we expect, where they're all children. But for the remaining hospitals, we do see a bit more variation, especially with UCLH, where we do see a mix of older and younger patients. Also with Lancashire, we tend to see that they typically have older patients compared to some of the other hospitals. And then lastly, we have the proportion of male patients for each of these drugs. So the proportions on the bottom, and then the drugs are on the y-axis. And you can kind of quite clearly see for the vast majority of these drugs, they're mostly male patients, with some of them being 75 to 100% male. So in summary, we see that the use of high-cost medicines varied a lot across the different hospitals, which could, I think, primarily be explained by differences in specialisms. Some UCLH especially is a specialist cancer centre for a lot of the rarer drugs, and there's a good chance maybe that a lot of patients are referred to UCLH from other hospitals, although we don't actually have that data linked, so we can't actually check. And this would also explain maybe why somewhere like Lancashire has a very small number of new users. Additionally, for the variation in age, again, could kind of be explained by specialisms. Obviously, Gosh would have younger patients, but it could also reflect the population in the region. So London would typically have a younger population than somewhere like Lancashire, which is a bit more rural. And the high proportion of male users, we could potentially explain that by the type of cancer that's being treated. So if it's prostate or breast cancer, you're going to see it skewed more one way or the other. But there's also some evidence to suggest maybe potential research bias in which that drugs that affect men more tend to get more research and the drugs get approved a bit quicker. But then also another possibility is that screening among male patients tends to be quite poor, attendance for screening, I mean, which can mean that men can sometimes present with more advanced stages of cancer, which would then maybe require some more extensive treatment. But yeah, that's all. I just thought it'd be a good way of showing the kind of work we've been doing with Heron. But yeah, thank you.
Fabulous, thank you, Ellen. That's really interesting, and thank you. So it's not been raised as a question, but Ethna, I don't know if you're willing to make your point as a question, or shall I do it on your behalf? Because you raise a really good point. And in Ethna's absence, I will raise the point that she has Oh, here we go.
Sorry, happy to raise it, but so as a hematology nurse by origin, I remember the origin story of rituximab and its use in myelomas and lymphomas, as you commented on. But in more recent years, there's been a move to use rituximab in rheumatology, such as arthritis and so on. So there's possibly a little bit of a confounder there that you need to maybe tap into and just double check on.
Okay, thank you. Yeah, I basically just went off ATC groupings to sort of classify these, but yeah, that's a good point. Thank you.
It is a really good point, and I think it's a general truth as well that these disease-modifier drugs, the biological therapies, are very likely to be used more widely than cancer use. So including a cancer diagnosis is quite important. And then, of course, you have the challenge of a cancer patient who's getting rituximab for their rheumatological condition. And that's quite hard at times to disentangle. But great point. Are there any other questions or comments for Ellen? And if not, and Danny, to give you a minute's warning, well, I'm going to move on to our last session, which is we've advertised on the agenda as a discussion, and in particular to talk through as a group through future plans and what should we, the UK-based cancer community who is interested in exploiting the value of OMOP, how should we take the next step? And can I pass that to you, Danny, to lead in your role as the head of the HERON network?
Sure, thank you so much, Geoff. And maybe also from the perspective of OHDSI UK as well. I think you all know I do a lot of international collaboration and work. So it's not only that I do work within the UK, but I think there is a unique opportunity in the UK for oncology research, given the nature of the NHS and the kind of continuity of care that we have in this country. I do think that there's unique challenges to oncology, not really specific to OMOP. I think most of the challenges I hear when people discuss the challenges with OMOP are, I always ask them, what is the counterfactual? What would you do if you didn't have OMOP, right? So I think there are challenges specific to this condition, but there are also lots of opportunities. There's a lot of funding, investment, lots of innovation, and there's, more importantly to me, a lot of need. So there's this increasing need to use real-world evidence in the oncology space because of those same specificities, right? So the increasing division of the condition into different phenotypes and finer cutting of one type of cancer into smaller pieces, which oncologists know better than I do. There's also, I think, increasing opportunities to do work on things that are not only medicines, so surgery, for example, surgical innovations, implants, and many other treatments that are not always medical. So I think we need to bear that in mind. And I think the UK will have to play a very important role in this space in Europe and internationally. As I mentioned earlier, I think the key challenge that we have nationally is that despite the NHS being, the kind of common link amongst all of us, each hospital trust pretty much has its own structure and an EHR system. So we need a way to interoperate. In the HERON UK collaboration, we are trying to essentially create a network of the willing who want to collaborate together and work together using OMOP. We have not much funding, but a lot of energy and a lot of willingness to make a difference. So if anyone who spoke today or anyone who has good quality data mapped to OMOP nationally and wants to participate in the HERON network, please get in touch. Let's talk and see how we can collaborate, because I think it would be fantastic if our studies could go beyond the current network. It's a bit of a catch-22 that because we don't have funding, we cannot onboard partners, but at the same time, the opportunities for funding will be better if we have more partners, right? So I think we need to find a way through. Specifically in oncology, there's I'm sure going to be many opportunities internationally from IHI, Horizon 2020, and the likes, where international partners will come to the UK and say, do you just want to work with one center? Do you just want to work with one hospital trust or one registry, or do you want to have access to data that has a greater coverage and provides the detail that you need? So I think we need to facilitate that. I'm not an oncologist. I've never been extremely interested in cancer research, but through my work in Optima, Eden, and in Darwin EU, I realized that this is an area where we can really make a difference. And I know that people like Geoff and others will be supporting this mission. So I'm really looking forward to seeing what we achieve.
So Danny, let me just respond to your point there about whether commercial organizations are interested in single center data, because historically they were, but I have to say that interest has waned to the point that I would actually argue it's probably now disappeared, that actually single center data is of less and less interest to organizations. And that's for two reasons. One, of all the biases that come from just looking at a single center. But secondly, the interest is increasingly on these ever rarer subtypes, the subdivisions of cancer. So we no longer get approached in leads for our data on lung cancer or breast cancer. We get asked about our data on triple negative breast cancer that contains a specific KRAS mutation. And invariably, we might have 11 of them. And no single center that's operating at a handful of or two handfuls of patients can contribute any useful information on its own. You have to do that in partnership with other centers within your region, your country, your nation, as in the 4 nations of the UK. But actually, I think for the increasingly rare subdivisions of uncommon cancers, I don't like pancreatic cancer, this is where you need multinational collaboration. And the common data model represented by OMOP is simply the only solution that I've discovered to these reproducible studies. So I think that's why I would be such an enthusiast for getting others to adopt an OMOP data model. And at one level, let's remember that if we were all dealing in OMOP, a cancer community in England was dealing in OMOP, we could do England studies. That would work absolutely fine. But we have the ability to do 4 nation studies, European and international studies as well. And therefore, a single common data model, I think, should be, core to what we do.
Yeah, thank you, Jeff. And I think Asia put it very well when she mentioned, you're going to need different types of data as well. So you're going to need the kind of registry data where you have certain level of granularity with larger numbers, but you're also going to need the hospital trust and hospital EHR where you have smaller numbers, but potentially more granularity. And I'm pretty sure that we're also going to be seeing a lot of work with biobanks of sorts, like, the UK Biobank, Genomics England, our Future Health that's starting soon. So all those pieces of data will be able to answer different questions alone. All of them are going to be relevant.
So I was hoping to put a question to Asia, but I think she may have dropped off. Is that right? And in an absence of her response, I'm going to assume that she has, because one of the very common questions that the research community would come back to me when I would suggest OMOP is that some of the data that we wish to analyze within this research project doesn't map onto an OMOP data model, and therefore we can't use OMOP. Danny, I wonder if you had any experience of both extending the OMOP data model and also the idea of using OMOP for 90% of your research data model, and then just adding different data items that can't be mapped to that, because that's a constant criticism that we get when we speak to others. Yeah.
Let me start by saying that is common criticism that comes from the oncology field and from any other field in medicine. If you talk to any rheumatologist, they will say, oh, but you don't have DAS28 or, BASDAI, whatever index. And again, this is all about counterfactuals. How will we do it otherwise, right? So the solutions are essentially 2 or 3. One is, as you say, you use mostly OMOP, but then you have an external piece of data. The only situation where I found myself doing that to now is with genomics. So we have done work where the genomic data was kept out of the OMOP CDM and curated to produce the bit of information we needed, just because it was so large and so difficult to move. And then only that bit of information that we needed was what we used then as a variable in the whole OMOP ecosystem. For everything else I've observed over these years, I've never been in a situation where something wasn't doable with OMOP. Most of the times when people say that, they don't really know the depth of the OMOP vocabularies and the OMOP domains. It's, they're probably coming from the perspective of someone who has been using a different data model or a single center data model, and they don't see the breadth and depth of OMOP. But if I'll tell you one situation where there was a new disease we had never dealt with before, where there was nothing on it in OMOP, and that was COVID. And I can tell you that when we started hearing of COVID in January 2020, February 2020, we obviously didn't have anything in the vocabularies relating to COVID because it didn't exist before. But over a few weeks, the vocabulary team, got themselves to work together and produce the concepts that we needed and the information that we needed. And we ended up publishing, I don't know how many papers on COVID, 40 or 50, including papers in the BMJ, the Lancet, you name it. So I don't think the OMOP CDM has that level of gap really in the structure. It's more a vocabulary issue that can stop you sometimes, and that can be fixed. What we, I think, need to learn is to prepare the OMOP CDM and to invest in it. I think when I explain to people what we do with OMOP, it's a commitment because this is a community effort. So if you are going to be using OMOP, you need to be a user, but also a contributor. You need to see yourself as someone who can say, actually, I am the expert in this type of breast cancer, and Danny, you don't have any idea about it. There's this mutation that you don't have in any of the vocabularies. And my answer would be, let's incorporate it then. The beauty of OMOP is that you can fix the vocabularies, improve them over the years with contributions. And then the final thing I will say, and then I will shut up for a bit. One of the key advantages of OMOP compared to other CDMs is that it preserves the source data. So if you have data in a source vocabulary that's not really there in the OMOP vocabularies, if you map in a way where you preserve your source concepts, you can always rescue them at the analytical stage. So that's a key advantage compared to other CDMs that people need to bear in mind, and most people don't remember.
And on that subject, one of the really interesting sort of extensions that, while it's news to me, and at one level, it may just be that I'm out of touch how long this has been possible, but there's an increasing move to get patient-reported data recorded and expressed within the OMOP data model. And there's a sort of parallel increase in the interest of patient-reported data for cancer outcomes. So at one level, that's a really useful extension that's going on. So Danny, can I pose another question? You've talked about limited funding being available through Heron UK. How do you think that the UK cancer community can take the next step? Is this something that centres and hospitals should be doing alone, or should there be a larger grant application being submitted to NIHR, CI UK, HDI UK, or even a consortium of pharma, of UK pharma? I mean, I know that with colleagues, we've talked about going through the ABPI to speak to a range of pharma companies, many of whom are highly interested in cancer data.
Yeah, I would say probably all the above. So Heron UK I see it as the seed for something much larger. I'll tell you what I would not do. I would not work in silos. I would not map my data on my own, put all the effort to then discover a year down the line that people have done things completely differently to mine. And then in the end, we cannot really use OMOP, as we should. So I think lots of the learnings from Heron will be about scientific coordination, direction, how we make decisions together. That's, I think, going to be the most important bit of information we'll get from Heron UK, rather than the specific bits of evidence that we will generate, which of course will be relevant as well. But I think each specific field will probably need a little bit of a different angle. And I am maybe naively hopeful that HDRS will invest in creating a bit of an infrastructure, not an infrastructure as in, a data lake, but infrastructure in terms of people
working together under some sort of direction so that they do things similarly. If that doesn't come from the HDRS, which in my opinion would be ideally suited for that, I think we will need to start writing grants to do this for different fields. So I heard just today that we've gotten a grant from NIHR program grants to do this for medical implantable devices. So you'll be bothered by some of the people in my team very soon, because we got that funding and we will want to do things. But the same should be true, I think, for oncology. I don't know what's the best funder for oncology, but if NIHR, UKHDRA, or industry are good funders for you, I think you should, go and do that. I think the European funds are also quite interesting for this. There's a few IHI calls in topic 12 that I think are relevant to this. I just learned today that unfortunately we are not eligible as UK institutions for call 12 topic 4, but there's 3 or 4 other topics where we could potentially make a difference. I think it's extremely important nationally and strategically that we follow as closely as possible the HDS developments, because otherwise we're going to be left behind. So I think there's a need for strategic investment in this space. And yeah, who should be funding that? As I said, ideally HDRS should be a key player in this, but if not, we will need to rally together and write lots of grants and get that funding in, because we will need the funding to make it happen. And then I am pretty optimistic that once the investment is done, this is quite self-sustainable, because there's going to be a massive need for this kind of data from all different stakeholders.
Yeah, I mean, there are people on the call which I won't name, but they may like to jump in, that some of the work that has been presented today is funded at a European level, but unfortunately on funding streams which exclude the UK. And that is explicitly very frustrating, that we can't partake in those studies. And the question is whether there are central funding sources, and HDRS is of course a new funding source in town, that in many ways can we use at one level their alignment to quite senior ministerial ambition to in many ways bias back into those European studies, because this stuff has to be done collaboratively. So
I can't agree more. I think this is a strategic decision that just needs to happen. We just need to work together to fund these studies. But we're going to need a network of specialist centers in the country that do a lot of cancer care and can work together. The same will be true for implantable devices. The same will be true for cardiovascular disease. And we just need this. It's like, I'm pretty sure that if you tell people that this doesn't exist, they would be surprised rather than anything else. I don't know, Geoff, if there's anyone else in the audience that might want to intervene, because I see a lot of messages written in the chat, but I will be very happy to hear people's voices and see people's faces.
Could we encourage anyone to come out of the chat and make their points in a more general forum? I mean, my personal hope is that we can get NHS England and the cancer registries from all 4 devolved nations to come together in this space. Because at one level, here we are talking about alignment with European countries, but at the moment, doing 4-nation cancer studies in the UK is challenging because of different data sets. So, Ethna, I can see your comment, but the connection to the cancer registries is essential, and I just couldn't agree more. I think the national cancer data sets just need to be accessible in OMOP form through the national secure data environments. But those are reasonably challenging ideas at the moment for reasons I don't completely understand. But we do have amazing cancer data in this country, but A, accessing it, and B, accessing it in standard form that aligns with other nations is particularly challenging. Thomas, can I entice you to ask your question?
Yeah, it wasn't necessarily a question. It was more like, I guess what I was just saying on the chat was like, how do we actually put in the foundations for the data side of things to make sure that we're able to contribute data either locally within local networks or at the national level or at the global level? Because at the end of the day, once you've got the data in the OMOP Common Data Model for oncology side of things, it's a scalability, it's a scale issue. And I think one of the things that we found with the Falcon network was that it's worked because the data partners are engaging with the network. And like we see it with Eden, on the Eden side of things, it's difficult because the money entices the Eden network, the data partners, and the data isn't necessarily updated versus, say, the Falcon network, where it's pretty much led by the oncology working group. And so how do we lean into those types of things to be able to deliver on, say, the Heron network, and to actually be able to scale these things out at a level that is possible with these types of things, regardless of where, like, who's contributing. I mean, obviously I work in pharma, but skill sets in data engineering and that side of things. So how do we know what resources we've got within the UK that could contribute to these things, and the lessons that we're learning from a global level and bringing them to the UK side of things as well? So it's also about kind of leveraging the resources that we have without necessarily engaging in the money side of things.
Yeah, absolutely. Izzy, I see you've turned your camera on. Do you have any thoughts or comments about UK funding within Europe?
I took your message, with the funding streams which were limited to member states, which, although the UK now is eligible for Horizon funds, the European Regional Development Fund is limited to member states, so excludes the UK and Norway for that matter, which are also within the DigiOne network that Stelios beautifully showcased some of the examples of the initiatives that we are leading. I had two comments. I mean, clearly, that's, there are pockets of money which are not available. So, it's the same. For example, these days, Horizon is becoming much more competitive because restricted funds in the US means that lots of US groups are applying for Horizon grants. So where Horizon grants were complicated, now we have also an additional stream of groups seeking those funds. So it's going to become even more challenging. But that's, I think is in line with what Danny mentioned and you were mentioning. We might have to find domestic funds which will bridge, as Danny said, if we lose the connection with the European health data space, and there is a new wave of funding coming in January, which are about these health data spaces, that will be hopefully the space of the HDRS, and hopefully that will take shape, but it's going to take some time. But that's clearly an obvious connection. But I was also thinking of two other comments that you've mentioned throughout the discussion. One of them was related, your question about at what level should OMOP take place, at the source, in a data lake, and that's a very key question in terms of, do you get the data already structured at the source and that is easier? And we had a number of conversations in terms of, how you could hijack the submission to COSD or the SDRS in Scotland, and that will give you a minimal data set, which will be already enabling you to do some of these across nations studies. And then there was also the question around, whether the question that you asked, Danny, in terms of whether everything should be in OMOP. And that's where you risk of duplicating. There are some data sources which are well structured, they are well established standards. If you try to squeeze them into OMOP, you will end up having an incomplete copy. And in terms of volumes, you will be those databases will be exploding. I'm thinking here of DICOM and all the radiology data. You have all the genomic data. If you will try to squeeze, getting a poor version of DICOM or a poor version of the how you map all the genomic data with VCF. So that's not a solution. The idea is the fairness, being able to query those data sources and, aggregate the data. So you don't have to replicate and have everything in a unique space. You need to have interoperability and being able to, add layers to your core data, and then you will be able to answer some of those questions. If you try to squeeze, you will end up having people trying to map those without perhaps the perfect tool skills. And then you will end up having with incomplete copies of data and huge volumes of big bills on your cloud services. So that's what I wanted to, some of the reactions to some of your comments, both Jeff and Danny.
Yeah, and I think there's opportunities for data minimization, and you don't need to have. I think specifically for things like imaging and genomics are very good examples, I should say, as you mentioned. So for genomics, you don't need to have the whole genome sequence in OMOP. I mean, what's the point? That has a very specific structure already that we have known for a few decades, right? I think the point there is if you have, and this is something we are experiencing, by the way, in the Darwin EU initiative as well, we now are unlocking access to multiple biobanks across Europe by essentially doing all the data curation, say building cohorts of drug users, building cohorts of people with certain outcomes, looking at eligibility criteria, measurements, anything you can imagine, using this exact same phenotypes in OMOP. And the only thing we import from external tables that live locally are the genotypes, right? So you are only exporting or importing really into the OMOP CDM 1 or 2 or 3 variables. I think this can scale up a little bit. It can go to, for example, I have an interest in pharmacogenomics where there's essentially 30 genes that are involved in the metabolism of most of the medicines we take or we administer. You could just have the information on those 30 in your OMOP CDM if that is of interest to, again, have one package of code that can do all the analysis. But I think we need to learn how to live in this world where we have 2 bits of information coming from different places, but most of the analysis, most of the curation happens in OMOP, and that facilitates your life dramatically. We're going to be having a lot of discussions with other colleagues around the world who have similar data, like all of us in the US has now got an OMOP. They collected the data in OMOP and it's now available in a cloud environment. We had someone from the VA visiting recently, my team, the Veteran Affairs in the US, where again, they have a 1 million people data set where they have GWAS data, like full genome sequence data. So there's a lot of opportunities that will be opened by OMOP, even if OMOP is only a good part, but not the entire picture. And I think the same is probably going to be true with oncology, given the advances in, immune therapies and gene therapies and many other things, right?
And maybe it's also learning from other communities. I remember the days where at some point it was impossible to have I'm thinking of genome browser Ensemble. And at some point, you will be able to deploy your features, but the number of SNPs the files were so huge that then the system evolved to have a sliding window. You didn't have to upload a text file with all the SNPs. You will keep it in the location and you will be able to stream. So that might be also maybe the possibility for DICOM or for genomics, not trying to even get in a slice of it. It is just having access for the particular question that you have. You're talking about pharmacogenomics, have a digital gene panel for those particular indications. You have a number of 10, 15 genes, and those will be the ones that you will be just looking into. So integrating slices of data as required without having to duplicate everything.
As the clock moves on to 3:45, I think we should call time on today's meeting, Danny, because I think we are at advertised time. So as I said at the beginning, this was a meeting I've been looking forward to enormously because it combines both the cancer and OMOP interest that I've had. And so I'm delighted the meeting's happened. I think it's been fabulous hearing the breadth and depth of experience from the speakers we've had. But thank you to everybody who's joined us this afternoon and this morning. I hope it's been of interest to others, and I hope that this acts as a springboard to a bigger and wider community working together on OMOP in cancer. So with that,
thank you, everyone.
Great to see you all. Thank you.